Inside the quest to reprogram immune cells into an anti-aging remedy. Add. Follow. Story by Miriam Fuzia, 1H. As the years wear down on us, our bodies accumulate a whole host of damaged cells that stubbornly refuse to die. Called senescent cells, these cellular oddballs are like the moldy fruit in the basket, hastening the spoilage of the rest or, in our body's case, ushering in a myriad of age-related diseases. In the quest for the fountain of youth, senescent cells have become a hot-ticket item. Scientists are actively researching the genes and other biological factors that make these cells so resilient, including testing a class of drugs called senolytics designed to wipe them out. Now, one group of researchers has figured out a way to reprogram a key player of the immune system to take down senescent cells like a heat-seeking missile of rejuvenation. The process involves genetically engineering white blood cells called T cells to create what's known as chimeric antigen receptor, or CAR, T cells. These specialized cells recognize and attack a specific cellular target. Researchers at Cold Spring Harbor Laboratory in New York tailored a CAR T cell to home in on a molecule found in large amounts of senescent cells called urokinase plasminogen activator receptor, UPAR. When these T cells were sicked on senescent cells in aged mice with metabolic issues and young mice fed a high-fat diet, which can trigger age-related senescence, the animals pulled a 180 with almost no side effects, losing weight, becoming more physically active, and seeing improvements in their metabolism. If we give it to aged mice, they rejuvenate. If we give it to young mice, they age slower. No other therapy right now can do this, Karina Amor-Vegas, the study's first author and an assistant professor at Cold Spring Harbor Laboratory, said in a press release. These findings were published Wednesday in the journal Nature Aging. Weaponizing T cells. In recent decades, CAR T cells have made a name for themselves in fighting cancer as a promising immunotherapy. They are often dubbed living drugs because the white blood cells can be taken directly from someone through a blood sample and then reintroduced after they've been genetically altered, which is lately done with the gene editing tool CRISPR. One significant challenge with CAR T cells, however, is finding just the right molecule or biomarker specific to the cell you want to target, a concern especially crucial in cancer treatment where you wouldn't want your living drug to target healthy instead of cancerous cells. No two cancer cells share the exact same cast of molecules on their cell surfaces, which is likewise for senescent cells. The new study relies on earlier research that laid the groundwork for that search. In a 2020 paper published in Nature, researchers from the Memorial Sloan Kettering Cancer Center, which included Amor-Vegas, found that senescent cells had more UPAR dotting their outside surfaces compared to other cell types. When the researchers tested anti-UPAR CAR T cells in two separate groups of mice, one with lung cancer and the other with liver fibrosis, a condition where healthy liver tissue becomes scarred and can lead to cirrhosis and liver cancer, the animals, surprisingly, lived longer. This success prompted a next step, testing whether a CAR T cell therapy could extend longevity in regular mice. Amor-Vegas and her colleagues used the same CAR T cells developed from their earlier experiment, intravenously infusing them into a group of mice between the ages of 18 to 20 months old, equivalent to 56 to almost 70 years old in humans. A control group didn't get the treatment. These mice were fed a normal diet, but, because of their advanced age, suffered from age-related metabolic dysfunction, a condition humans also develop as we age, where they had elevated blood sugar levels and weren't able to move around or exercise all that much. CAR T cells targeting UPAR were also given to a group of much younger mice, about 3 months old, before they were started on a high-fat diet, where about 60% of their calories came from fat. Studies show that consuming that amount of fat can promote senescent cells through the stress and inflammation caused by obesity. While these animals didn't yet have any metabolic disorders, the CAR T cells were given as a prophylactic in an attempt to delay the inevitable aging. For both groups of mice, the CAR T cells did their magic. Older mice found themselves healthier with lower glucose and insulin levels and fewer inflammatory markers circulating in their blood. They were moving around and weighed less. The younger mice who were given the treatment about a month and a half before starting their high-fat diet didn't gain as much weight, and their blood sugar levels were better compared to their counterparts who didn't get this treatment. Further research needed. These findings are striking in that we have a possible new path to use our own immune cells to shed the cellular damage wrought by aging that could be long-lasting even after one dose. For example, in the younger mice, the researchers found that the anti-UPAR T cells were still hanging around in the animal's spleens and livers and were mostly a subtype of T cells, known as a CD8 plus T cell, that has the ability to fight off harmful cells. T cells have the ability to develop memory and persist in your body for really long periods, which is very different from a chemical drug, said Amor Vegas. With CAR-T cells, you have the potential of getting this one treatment, and then that's it. For chronic pathologies, that's a huge advantage. Think about patients who need treatment multiple times per day versus you get an infusion, and then you're good to go for multiple years. However, implementing an anti-aging CAR-T cell therapy needs to go through more clinical trials with both animals and eventually humans before it can ever reach the market. The good news is that research into engineering and developing CAR-T cells is pretty advanced, and there are more clinical trials, specifically for cancer, now than ever. As plans for cheaper and even off-the-shelf CAR-T cell therapies are being explored, your anti-aging, living drug could be here sooner than you think. Nanclustering/science photo library/science photo library/Getty Images copyright provided by Inverse. Inside the quest to reprogram World health leaders warn of pandemic 20 times worse than COVID. Add. Follow. Story by Stephanie Whiteside, 15H. Health topics mentioned in this article. Valley fever. Ebola. Fever. News Nation. At the recent World Economic Forum, the World Health Organization issued a warning to world leaders, saying the world could face a pandemic 20 times worse than COVID-19 in the future. Scientists call it Disease X, a term that recognizes the next global pandemic could come as the result of an unknown pathogen rather than the spread of a currently recognized disease. Scientists with the WHO held a session last week calling on world leaders to work together to develop strategies to prevent or manage a Disease X pandemic in the future. Disease X is not real. It is the name given to a hypothetical pathogen that is being used to help plan for future health crises. Global healthcare experts on Wednesday spoke on a WF panel called Preparing for Disease X. The name was coined by the World Health Organization in 2018. One of the issues raised was developing better communication strategies to reduce misinformation and conspiracy theories, even as some took to X, formerly known as Twitter, to call the session itself a conspiracy against freedom. Ukrainian soldiers developing mouse fever. Scientists say the most likely culprit for Disease X would be a respiratory virus, possibly one already circulating in animals that hasn't yet made the jump to humans. Without preparedness, the WHO warned, a pandemic from Disease X could cause much more damage than COVID, which has killed more than 7 million worldwide. The WHO has already begun some initiatives to protect against a future pandemic, including efforts to support technology sharing and boost disease surveillance between countries. While Disease X was the focus of the session, it's not the only illness that concerns epidemiologists. Other viruses that could potentially cause a pandemic include Ebola, Marburg, Crimean Congo hemorrhagic fever, Lassa fever, SARS, MERS, Nipah virus, Rift Valley fever, Zika virus, and new evolutions of COVID-19. The Associated Press contributed to this report. Copyright 2022 Nexstar Media Incorporated. All rights reserved. This material may not be published, broadcast, rewritten, or redistributed. The regenerative medicine revolution. What if aging was optional? With the advances in regenerative medicine, it could be. By using your own cells to restore and repair damaged tissue, you could slow down the aging process and maintain your youthful appearance and health. Imagine a world where you could choose to age gracefully or stay young forever. The possibilities are just an injection away. Think of how a starfish can regenerate a limb, or how an amphibian salamander can do the same. These creatures are much lower on the evolutionary ladder than humans, yet they have this pre-programmed ability. Certainly, there is further potential in the human body for the same. And this is what we are seeing. Regenerative medicine is not just on the horizon, medicine or something of the future. We are seeing advances in all sorts of cellular technologies, immunotherapies, gene therapies, and stem cell therapies. And many of these treatments are available for use today. This interdisciplinary field of science focuses on the use of stem cells, gene therapy, and other advanced techniques to repair, replace, or regenerate damaged or diseased cells, tissues, and organs. Regenerative medicine is revolutionizing the way we think about aging and health, and it is providing new hope for those suffering from age-related diseases and conditions. This revolutionary field of medicine has the potential to treat a wide range of conditions, from heart disease and cancer to spinal cord injuries and Alzheimer's disease. It is helping athletes get back to their sport without the side effects of medication, and it's getting weekend warriors back to normal using natural cell-based therapies. Stem cells have been showing great promise. What was once a controversial issue due to the use of embryonic stem cells is now widely accepted through the use of non-embryonic adult somatic cells. Nearly every adult tissue in the adult body has some stem cell source. They be harvested from a variety of sources, including the patient's own fat tissue, bone marrow, and even from hair follicles. But other ethical neonatal sources are also readily available from umbilical cord blood, placental tissue, and other sources, safely using tissue that would otherwise be thrown away to help pave the way towards scientific progress. The regenerative medicine revolution is also making it possible to use gene therapy to treat a variety of diseases. Gene therapy involves introducing new genes into cells to replace defective ones. This can be used to treat genetic diseases, such as cystic fibrosis, as well as to help the body fight off cancer and other diseases. Very soon, some of our most tragic diagnoses will simply be a DNA splice away from a complete cure. Through bioprinting and tissue engineering, scientists are also developing new organs and tissues. Tissue engineering involves combining cells, scaffolds, and other materials to create replicate models of those tissues which are biologically active. The result is the engineering of new hearts, livers, and other organs, as well as the ability to repair damaged organs. Perhaps the most studied and employed type of regenerative technology is the use of platelet-rich plasma for healing joints, tendons, and ligaments. Platelet-rich plasma is made by taking a portion of a patient's blood and through a process of centrifuging the blood, the red blood cells are separated from the platelets which can then be concentrated. The concentrated platelets can then be activated which induces them to release powerful growth factors into the surrounding environment. Clinical studies have shown that PRP injections are of much benefit to areas in need of repair, such as the elbow, wrist, shoulder, hip, or knee tendons and joints. And when you feel improvement from a regenerative treatment, you can be certain that the improvement is from repair, unlike steroids and other pain medications designed to simply reduce swelling and chronic pain. Ryan Welter, MD, PhD, is the CEO of Regeneris Medical. Arctic zombie viruses. New pandemic threat warned by scientists. Ad. Stormshot new online adventure game. Ad. Casual men choose the best men's shoes for walking and standing all day. Follow. Story by Sania, 20H. People are worried about a strange new Arctic zombie viruses, and scientists are warning about it. They say that very old viruses, frozen in the Arctic ice for thousands of years, could wake up because of global warming. If they do, we could have big diseases spreading across the world. Looming threat of Arctic zombie viruses. Jean-Michel Claverie, a genetics expert from Aix-Marseille University, is leading the research. He says, there are viruses up there with the potential to infect humans and start a new disease outbreak. For now, scientists study diseases that start in the southern parts of the world and move to the north. They aren't paying much attention to possible diseases that could start in the north and move southwards. That's something we need to think about, Claverie explains. To deal with that, scientists plan to form a network to monitor the Arctic region. If ancient viruses cause any disease outbreak, they could spot it early and provide medical help. That will stop infected people from leaving the region and spreading the disease. A closer look into Arctic zombie viruses and their potentials. Marion Koopmans, a virus expert from the Erasmus Medical Center, also worries about the possibility. She says, we don't know what viruses are lying out there in the permafrost, but there's a real risk that one might cause a disease outbreak. We have to be prepared for this. The zombie viruses frozen in Arctic ice are really old, buried there for thousands of years. Claverie's team of scientists had studied some of them in 2014. They found that the viruses were still able to infect single-cell organisms. The viruses we isolated posed no risk to humans. But it doesn't mean that other frozen viruses can't cause illnesses in humans, said Claverie. He also highlighted that scientists found traces of human pathogens like pox viruses and herpes viruses in the permafrost. Permafrost is really cold and without any air, which can keep things alive for a long time. It covers 1/5 of the Earth's northern half, and some of it has been frozen for thousands of years. Claverie also explained that the world's permafrost layers are not the same anymore. The ones in Canada, Siberia, and Alaska are melting because of climate change. This change could wake up the old viruses in the permafrost. Also, movements of ships and industrial works in Siberia are increasing because of the melting Arctic ice. Big mining operations could dig into the deep permafrost layers, releasing the zombie viruses. Workers may breathe in these zombie viruses, which could lead to a health disaster. Koopmans emphasized, epidemic outbreaks usually start when there's a change in land use. We could witness a similar thing in the Arctic, which could be dangerous. The viral threat from permafrost is real and could be very scary. The deepest layers of permafrost might have viruses that are hundreds of thousands of years old, even older than a human species. The scientists say that we have never have come into contact with these viruses, so our bodies might not know how to fight them. As Claverie says, although unlikely, the scenario of an old virus infecting a Neanderthal coming back to infect us has become a real possibility. Thus, scientists think we should be ready for these zombie viruses. The threat is real, and we need to be prepared for it. Arctic zombie viruses. New pandemic threat warned by scientists. Copyright provided by Sportskeeda. Scientists are monitoring the Arctic region. Image via Unsplash/Davide Cantelli. Copyright provided by Sportskeeda. These zombie viruses have been buried for thousands of years. Breakthrough in DNA repair. Researchers reveal secret of Rika's precision. Add. Follow. Story by Rizwan Chowdhury, 3D. DNA is the blueprint of life, but it is constantly under attack from various sources of damage. The DNA is constantly exposed to various sources of damage, such as UV rays, chemicals, or errors during replication. One of the most serious types of damage is a double-strand break, where both strands of the DNA helix are severed. This can lead to cell death or mutations that cause diseases like cancer. Homologous recombination. Fortunately, nature has a way of fixing these breaks through homologous recombination, HR. This process involves a protein called RICA, or its equivalent in different organisms, that binds to a single-stranded end of the broken DNA and searches for a matching sequence in an intact double-stranded DNA nearby. Once it finds the match, it inserts the single strand into the double helix and repairs the break using the intact DNA template. This process is essential for maintaining the integrity and diversity of DNA, but how exactly does RICA find the right place to insert the single strand? Does it unwind the double strand to expose the matching sequence, or does it somehow slide along the helix without disturbing it? Researchers from Tokyo Metropolitan University and Tokyo Metropolitan Institute of Medical Science have solved this mystery by testing two competing HR models. They used a mutant version of RICA that could not unwind the double strand and measured the amount of twisting in the DNA during the process. They found that RICA does not unwind the double strand during the search phase, but only when it inserts the single strand into the helix. This means that RICA can find the match without disrupting the structure of the DNA, which is remarkable considering the complexity and variability of DNA sequences. This discovery sheds new light on the mechanism and efficiency of HR, which has implications for cancer research. For instance, some genes linked to breast cancer, such as BRCA1 and BRCA2, are loading the single-stranded DNA onto RICA, or its human counterpart, RAD51. If these genes are defective, HR may fail or become error-prone, leading to more DNA damage and cancer risk. By understanding how RICA works, the researchers hope to find new ways to prevent or treat cancer caused by faulty HR. The study was published in the journal Nucleic Acids Research. The Japan Society supported it for the promotion of science and the Japan Agency for Medical Research and Development. Study abstract. RICA protein and RICA/RAD51 orthologs are required for homologous recombination and DNA repair in all living creatures. RICA/RAD51 catalyzes formation of the D-loop, an obligatory recombination intermediate, through an ATP-dependent reaction consisting of two phases: homology recognition between double-stranded DS DNA and single-stranded SS DNA to form a hybrid duplex core of 6 to 8 base pairs and subsequent hybrid duplex/D-loop processing. How dsDNA recognizes homologous ssDNA is controversial. The aromatic residue at the tip of the beta hairpin loop, L2, was shown to stabilize dsDNA strand separation. We tested a model in which dsDNA strands were separated by the aromatic residue before homology recognition and found that the aromatic residue was not essential to homology recognition, but was required for D-loop processing. Contrary to the model, we found that the double helix was not unwound even a single turn during search for sequence homology, but rather was unwound only after the homologous sequence was recognized. These results suggest that dsDNA recognizes its homologous ssDNA before strand separation. The search for homologous sequence with homologous ssDNA without dsDNA strand separation does not generate stress within the dsDNA. This would be an advantage for dsDNA to express homology-dependent functions in vivo and also in vitro. Breakthrough in DNA repair. Researchers reveal secret of RICA's precision copyright provided by Interesting Engineering. Two competing models exist for homologous recombination. The results of this study support a model where the double-stranded DNA is not unwound during the homology search. Tokyo Metropolitan University copyright provided by Interesting Engineering. Related video: Cancer experts unveil groundbreaking discovery, eliminates need for invasive surgeries. WON. Chinese scientists test chilling new COVID Frankenvirus with 100% kill rate in humanized mice. Chinese scientists experimented with a mutant COVID strain that has a 100% kill rate in humanized mice. The lethal virus attacked the brains of the mice that were engineered to have a similar genetic makeup to humans, according to the new study published this year. 2. Workers seen next to a cage with mice inside the lab in Wuhan, China. Credit AFP. 2. The deadly virus infected the lungs, bones, eyes, tracheas, and brains of the dead mice. Credit Getty. All the mice that were infected with the virus, known as GX_P2V, died within 8 days. Researchers noted it was a surprisingly rapid death rate, The virus infected the lungs, bones, eyes, tracheas, and brains of the dead mice. And the infection of the brain was strong enough to kill the human-like mice. In the days before their deaths, the mice lost weight, had a hunched posture, and moved sluggishly. And their eyes turned completely white the day before they died, the researchers in Beijing said. The study is the first of its kind to report a 100% death rate in mice infected by the COVID-related virus. Scientists said it underscores a spillover risk of GX_P2V into humans. The virus was a mutated version of GX/2017, a coronavirus-related pathogen that was reportedly discovered in Malaysian pangolins in 2017. Francois Ballou, an epidemiology expert at UCL's Genetics Institute, slammed the research as reckless. I can see nothing of vague interest that could be learned from force-infecting a weird breed of humanized mice with a random virus, the professor said. Conversely, I could see how such stuff might go wrong. The preprint does not specify the biosafety level and biosafety precautions used for the research. The absence of this information raises the concerning possibility that part or all of this research, like the research in Wuhan in 2016 to 2019 that likely caused the COVID-19 pandemic, recklessly was performed without the minimal biosafety containment and practices essential for research with the potential pandemic pathogens. Dr. Gennady Glinsky, a retired professor of medicine at Stanford, said, "This madness must be stopped before too late." The study doesn't appear to have any ties to the Wuhan Institute of Virology. The Wuhan lab has been at the center of the lab leak theory ever since COVID first emerged just a stone's throw from the facility, which was known to be studying very similar bat viruses. Many scientists and intelligence officials suspect bungling researchers at the lab accidentally spread COVID during risky experiments on bat coronaviruses. Even the FBI and the U.S. Department of Energy now believe COVID most likely leaked from a lab in China. Documents previously uncovered by Drastic, a team of scientists and sleuths investigating COVID's origins, revealed how U.S. scientists applied for funding to create a COVID-like virus in China. A year later, COVID-19 emerged in Wuhan and spread across the world, killing nearly 7 million people. Emails obtained by U.S. Right to Know, USART-K, show how U.S. scientists appeared to cover up their plan to carry out the high-risk coronavirus experiments in Wuhan with shoddy biosafety measures. The Sun also revealed that U.S. government health officials deliberately downplayed a lab leak as the most likely cause of the pandemic. Former intelligence chief John Ratcliffe and top health department official Dr. Robert Kadlec spoke out about the fact what was being said in public was not supported by their own intelligence. Over the summer, reports suggested that U.S. spies were probing whether COVID was created by a Chinese military scientist before it leaked from a Wuhan lab. Zhou Yusen, who worked for the People's Liberation Army, filed a patent for a COVID vaccine before the pandemic was declared, and mysteriously died just weeks later. It followed a secret memo that revealed that China ordered scientists to destroy all early coronavirus samples from their labs just 2 days after the world was first told about COVID. As the pandemic was starting to explode across the world, panicked Beijing ordered the labs to share their samples with the government or destroy them on the spot, the leaked order shows. BrainNet, a multi-person brain-to-brain interface for direct collaboration between brains. Abstract. We present BrainNet, which, to our knowledge, is the first multi-person non-invasive direct brain-to-brain interface for collaborative problem solving. The interface combines electroencephalography, EEG, to record brain signals and transcranial magnetic stimulation, TMS, to deliver information non-invasively to the brain. The interface allows 3 human subjects to collaborate and solve a task using direct brain-to-brain communication. Two of the three subjects are designated as senders, whose brain signals are decoded using real-time EEG data analysis. The decoding process extracts each sender's decision about whether to rotate a block in a Tetris-like game before it is dropped to fill a line. The sender's decisions are transmitted via the internet to the brain of a third subject, the receiver, who cannot see the game screen. The sender's decisions are delivered to the receiver's brain via magnetic stimulation of the occipital cortex. The receiver integrates the information received from the two senders and uses an EEG interface to make a decision about either turning the block or keeping it in the same orientation. A second round of the game provides an additional chance for the senders to evaluate the receiver's decision and send feedback to the receiver's brain, and for the receiver to rectify a possible incorrect decision made in the first round. We evaluated the performance of BrainNet in terms of 1. Group-level performance during the game, 2. True/false positive rates of subjects' decisions, and 3. Mutual information between subjects. 5 groups, each with 3 human subjects, successfully used BrainNet to perform the collaborative task, with an average accuracy of 81.25%. Furthermore, by varying the information reliability of the senders by artificially injecting noise into one sender's signal, we investigated how the receiver learns to integrate noisy signals in order to make a correct decision. We found that, like conventional social networks, BrainNet allows receivers to learn to trust the sender, who is more reliable, in this case, based solely on the information transmitted directly to their brains. Our results point the way to future brain-to-brain interfaces that enable cooperative problem solving by humans using a social network of connected brains. Data availability. Experiment data and code are available upon request. References: Rao, R. P. N. et al. A direct brain-to-brain interface in humans. PLOS 19, 1-12. https://doi.org/101371/journal.pwn 0111332 2014. Article SAS Google Scholar. Rao, R. P. N. et al. Direct brain-to-brain communication in humans, a pilot study. http://homes, C.est. Washington, edu/tilde/Rao/brain2brain. Access January 27, 2019, 2013. Min, B. K. J. Marzelli, M. and U. S. S. Neuroimaging-based approaches in brain-computer interface. Trends in Biotechnology 28, 552-60, 2010. Article SAS Google Scholar. Grau, C. et al. Conscious brain-to-brain communication in humans using non-invasive technologies. PLOS 19, 1-6. https://doi.org/101371/journal.pwn 0105225 2014. Article MathSciNet SAS Google Scholar. Stocko, A. et al. Playing A new COVID variant is dominant in the US. Know these symptoms. The United States is in the middle of a winter wave of COVID-19 driven by a heavily mutated, fast-spreading new variant called JN.1. The highly contagious Omicron subvariant now accounts for over 85% of cases nationwide. JN.1 is a direct offshoot of BA.2.86, aka Parola, with an additional mutation that affects its ability to evade immunity. In December, JN.1 rapidly overtook other variants, including HV.1 and EG.5 or ERIS, to become the dominant strain in the US and globally. The JN.1 variant may be intensifying the spread of COVID-19 this winter, the U.S. Centers for Disease Control and Prevention said in an update on January 5th. According to some experts and data models, the U.S. is in its second-largest COVID wave, smaller than only the Omicron surge in late 2021 and early 2022. Low vaccine uptake, waning immunity, and holiday gatherings likely also fueled the recent spike in cases. Currently, JN.1 is the fastest-growing variant in the U.S., per the CDC. In the last month, the share of cases caused by JN.1 has quadrupled. During a two-week period ending on January 20th, JN.1 accounted for 85.7% of cases in the U.S., per the CDC's latest estimates. This was a steep increase from 62% during the previous two-week period ending on January 6th and 44% during the two-week period ending on December 23rd. In November, JN.1 accounted for less than 5% of cases. JN.1's takeover comes as COVID hospitalizations rise, influenza continues to spread, and RSV activity remains high in many places across the country. What are the symptoms of JN.1? Is it more transmissible, and does it affect tests and vaccines? Here's what experts know about JN.1 so far. What is JN.1, the new COVID-19 variant? JN.1 was first reported in August 2023, and it has spread to at least 41 countries so far, according to the World Health Organization. On December 18th, the WHO classified JN.1 as a variant of interest due to its rapid spread worldwide. Just like other COVID-19 strains that gained dominance in the last year, JN.1 is part of the Omicron family, which emerged in late 2021. Think of the variants as children and grandchildren of Omicron. They're part of the same extended family, but they each have their own distinctive personalities, Dr. William Schaffner, professor of infectious diseases at Vanderbilt University Medical Center, tells Today.com. JN.1 descended from BA.2.86, which is a sublineage of the Omicron BA.2 variant, Today.com previously reported. That's what sets JN.1 and BA.2.86 apart from the other prevailing variants like HV.1 and EG.5, which descended from Omicron XBB. BA.2.86 has more than 30 mutations compared to the Omicron XBB.1.5 variant, the dominant strain for most of 2023 and the variant targeted in the updated COVID-19 vaccine, Today.com previously reported. When its parent BA.2.86 emerged, everybody was worried because it had a lot of mutations and looked like it was going to evade a lot of the immunity from vaccines and infection in the population, Andrew Pekosh, Ph.D., professor and vice chair in the Department of Molecular Microbiology and Immunology at the Johns Hopkins Bloomberg School of Public Health, tells Today.com. But BA.2.86 sort of fizzled out, he adds. Laboratory data suggests that Parola is less contagious and immune-evasive than scientists once feared, NBC News reported. JN.1, however, picked up an additional mutation in its spike protein, says Pekosh. Spike proteins help the virus latch onto human cells and play a crucial role in helping SARS-CoV-2 infect people, per the CDC. This mutation may affect JN.1's immune escape properties, says Pekosh. Now it's circulating and growing at a really fast rate compared to other variants, as well as the parent it's derived from, BA.2.86, says Peacock. In early November, JN.1 accounted for fewer than 1% of COVID-19 cases in the US. Several weeks later, it was driving over 20% of cases, Dr. Michael Phillips, chief epidemiologist at NYU Langone Health, tells Today.com. Now, it's causing the majority of cases worldwide. However, JN.1 does not pose an increased public health risk compared to other variants in circulation, the CDC and WHO said. What are the symptoms of JN.1? It's not known whether JN.1 causes different symptoms from other variants, according to the CDC. Right now, there's nothing that says that JN.1 infection is any different from previous COVID variants in terms of disease severity or symptoms, but we're paying close attention, says Peacock. The symptoms of JN.1 appear to be similar to those caused by other strains, which include sore throat, congestion, runny nose, cough, fatigue, headache, muscle aches, fever or chills, loss of sense of taste or smell, shortness of breath or difficulty breathing, nausea or vomiting, diarrhea. According to the CDC, the type and severity of symptoms a person experiences usually depends more on a person's underlying health and immunity rather than the variant which caused infection. While severe infections do still occur, overall, COVID-19 is causing a lot of milder illness, says Schaffner. Some doctors have reported that upper respiratory symptoms seem to follow a pattern of starting with a sore throat, followed by congestion and a cough, NBC News previously reported. The virus is adapting. I think it's getting better at infecting humans and evading pre-existing immunity in the population, but it's not changing symptomology too much, says Peacock. At this time, there's no evidence that JN.1 causes more severe infection, the experts note. Is JN.1 more transmissible? One of the things these Omicron variants have in common is that they are highly contagious, and as new variants crop up, they seem to be as contagious or even more contagious than the previous variants, says Schaffner. According to the CDC, the continued growth of JN.1 suggests that the variant is either more transmissible or better at evading our immune systems. JN.1 is likely more transmissible than its parental virus because we've seen an increase in case numbers that we didn't with BA.2.86, says Peacock. Many of the newer variants, including JN.1, have another mutation that affects how strongly the spike protein binds to cells in the respiratory tract, says Peacock. We know that it's probably helping the virus become better at replicating and helping the virus evade more of that pre-existing immune response, he adds. Over 97% of people have natural or vaccine-induced antibodies against the SARS-CoV-2 virus, the CDC said, but this immune protection fades over time. It's too early to tell exactly how JN.1's transmissibility or immune escape properties compare to other Omicron variants, but it's clear that JN.1 is very virulent, experts say. The current strain right now seems to be packing a meaner punch than the prior strains, Dr. Joseph Cabaza, a pulmonary and critical care specialist at the Cleveland Clinic, previously told Today.com. Will JN.1 cause a surge? Right now, JN.1 is increasing in terms of the percentage of COVID-19 cases it's causing, and there's also been an increase in total cases, says Peacock. Test positivity, an early indicator of case levels, is also on the rise, says Phillips. The rate was 11.8% during the week ending on January 13th, per the CDC. The CDC no longer tracks the total number of cases in the U.S. Wastewater data published by the CDC show that the viral activity level for COVID-19 is currently very high nationally. Now, because it is more widespread, it's also reaching older people, people with chronic underlying conditions, those who are immune compromised, and sometimes the very young, and those are the most vulnerable, says Schaffner. Hospitalizations increased by 6.5% and intensive care unit admissions by 12.9% during a two-week period ending on January 13th, according to an NBC News analysis. The good news is that as of yet we're not seeing severe disease or hospitalizations going up significantly, and ICU admissions are still low, but we're going to watch these carefully, says Phillips. According to Schaffner, the latest COVID-19 trends are not indicative of a massive surge, but rather expected seasonal increases. Experts anticipated COVID-19 activity to rise around this time as the U.S. entered its winter and respiratory virus season. In recent years, COVID-19 has followed a pattern of increasing and peaking around New Year, according to the CDC. It's unclear how this latest wave driven by JN.1 will pan out in the coming months, but at the present time, it's nothing like what COVID-19 has done to us in previous years, says Schaffner. Do COVID-19 tests detect JN.1? All COVID-19 diagnostic tests, including rapid antigen tests and PCR tests, are expected to be effective at detecting JN.1 as well as other variants, according to the CDC. Testing is an important tool to protect yourself and others from COVID-19, especially ahead of indoor gatherings, says Schaffner. The symptoms of COVID-19 are often indistinguishable from those caused by other viruses spreading right now, the experts note. These include respiratory syncytial virus, RSV, influenza, and rhinovirus, which causes the common cold. The experts urge anyone who becomes ill or is exposed to COVID-19 to take a test, especially people at higher risk of severe disease, such as people over the age of 65 who are immunocompromised and who have underlying health conditions. Every American can order 4 free at-home COVID-19 tests from the government, which will be delivered by mail via the U.S. Postal Service. To order your free tests, go to covitus.gov. Get tested because, whether it's COVID or flu, we have treatment available, says Schaffner. Current treatments are also expected to be effective against JN.1, the CDC said. JN.1 should be just as sensitive to the antivirals available as any other variants, says Pekosh, adding that antivirals like Paxlovid are most effective when taken within the first few days after infection. Does the new COVID vaccine protect against JN.1? The new, updated COVID-19 vaccines, recommended for everyone 6 months and older, are expected to increase protection against JN.1 as well as other variants, the CDC said. Although the shots target Omicron XBB.1.5, which has since been overtaken by HV.1, JN.1, EG.5, and others, there is still evidence that it will protect against new strains circulating this winter, Today.com previously reported. Data from laboratory studies show that the vaccine appears to generate a strong immune response against JN.1's parent strain, BA.2.86, Schaffner notes. The new vaccines also protect against severe disease, hospitalization, and death, the experts emphasize. So even if you get COVID-19 after vaccination, the infection will likely be milder and it can keep you out of the hospital, Phillips adds. However, uptake of the new booster among the U.S. population has been low so far, the experts say. As of January 19th, only about 21% of adults and 11% of children in the country have gotten the updated vaccine, according to the latest CDC data on vaccination trends. Last month, CDC officials issued an alert to warn about low vaccination rates against COVID-19, flu, and RSV in the U.S. Now is the best time to get vaccinated if you haven't already, the experts say. The sooner you get vaccinated, the sooner you'll be protected, and it does take 7 to 10 days for protection to build up to the maximum, says Schaffner. Phillips recommends everyone, especially high-risk individuals, to get the seasonal influenza shot as well. Hospitalizations for flu increased 200% over the past month, according to the CDC's December 14th warning. Getting vaccinated is the best present you can give yourself and your family, Schaffner adds. How to protect yourself from JN.1. Every day, but especially during respiratory virus season, people can take steps to protect themselves and others from COVID-19. The experts encourage everyone to stay up to date with COVID-19 vaccines, test if you have symptoms, isolate if you have COVID-19, avoid contact with sick people, improve ventilation, wear a mask in crowded, indoor spaces, wash your hands with soap and water. This article was originally published on Today.com. When and how could humans talk to animals? Add. Follow. Story by Devika Rao Foramo. We have yet to overcome the communication barrier between humans and animals, but we may be getting closer by the day. How could we talk to animals? The key to understanding animal language could be artificial intelligence, AI, according to experts. The emergence of powerful AI algorithms, and specifically large language models, has made researchers more hopeful in communicating across species, explained an essay published in the journal Current Biology. However, there are some areas of difficulty in navigating the task. The large language model would have to use the animal's own communicative signals in a variety of contexts and produce a measurable response to it as if it were communicating with a conspecific and not a machine. Animals communicate very differently than humans, and we tend to believe that what we cannot observe does not exist, because our sense of hearing is relatively weak compared to other species, Karen Backer, professor at the University of British Columbia, told the Australian Broadcasting Company, ABC. There's a lot of communication in nature that simply passes us by. Developing animal communication requires that we stop anthropomorphizing and comparing animal communities to human, umwelt, which is the lived experience of organisms, Backer told Scientific American. In order to immerse in a certain species' communication style, researchers are turning to digital bioacoustics, which relies on very small, portable, lightweight digital recorders to detect patterns in non-human communication without the disruption that comes from introducing human observers in an ecosystem. Have we made any headway? Scientists have made a number of fascinating discoveries regarding animal communication recently. They discovered that dolphins call each other by names, that mothers speak to their young in motherese, and honeybees use body movements and sound to communicate. In the case of the honeybees, researchers have used computer models and AI algorithms to be able to track individual bees, and they're able to determine what impact the communication of an individual might have on another bee, Backer said. From that emerges the ability to decode honeybee language. Scientists took the next step in creating a robo-bee that could enter the hive, and it would essentially emit commands that the honeybees would obey. The experiment was only successful once, but proved to be a huge step forward. This is slowly opening our minds not only to the wonderful sounds that non-humans make, but to a fundamental set of questions about the so-called divide between humans and non-humans, our relationship to other species. These types of discoveries can help humans understand animals, which can have larger implications. The point is not really to talk to animals, the point is to understand them, said Ozza Raskin, co-founder of the Center for Humane Technology and the Earth Species Project, per Axios. When this becomes the thing that the entire world sees, suddenly I think we can accelerate and be a force multiplier for every other conservation and climate action out there. What are potential implications? The ability to communicate with and understand other species could be monumental in shaping our collective environmental knowledge. Understanding what animals say is the first step to giving other species on the planet a voice in conversations on our environment, Kay Firth Butterfield, the World Economic Forum's head of AI and machine learning, told Popular Mechanics. Communication with animals could aid conservation efforts, transforming our relationship with nature. With this progress, we anticipate that we are moving rapidly toward a world in which two-way communication with another species is likely. But this progress also raises a number of philosophical and ethical questions. "We're assuming other species want to talk to us," remarked Raskin, especially if the animals realize they are not speaking to one of their own. In addition, this technology could be a Pandora's box potentially used to increase poaching and other destructive measures. Advanced bioacoustic technologies could be used to exploit animals that haven't previously been domesticated by humans, prompting a whole new world of control, wrote ABC. Animal communication requires understanding animals' languages in their context. Copyright Getty Images. Related video. Do humans need to hibernate too? New York Post. View on watch. Continue reading. Ad. Ad. Sponsored content. Trusted Board Portal Software, Governance Management Platform. Animal communication rapidly toward a world when and this emerging brain stimulation technology could boost cognitive function. Ad. Follow. Story by The Conversation and Shrey Grover, 8MO. Health topics mentioned in this article: dementia, psoriasis, bipolar disorder. Figuring out how to enhance a person's mental capabilities has been of considerable interest to psychology and neuroscience researchers like me for decades. From improving attention in high-stakes environments, like air traffic management, to reviving memory in people with dementia, the ability to improve cognitive function can have far-reaching consequences. New research suggests that brain stimulation could help achieve the goal of boosting mental function. In the Reinhardt Lab at Boston University, my colleagues and I have been examining the effects of an emerging brain stimulation technology, transcranial alternating current stimulation, or TACS, on different mental functions in patients and healthy people. During this procedure, people wear an elastic cap embedded with electrodes that deliver weak electrical currents oscillating at specific frequencies to their scalp. By applying these controlled currents to specific brain regions, it is possible to alter brain activity by nudging neurons to fire rhythmically. Why would rhythmically firing neurons be beneficial? Research suggests that brain cells communicate effectively when they coordinate the rhythm of their firing. Critically, these rhythmic patterns of brain activity show marked abnormalities during neuropsychiatric illnesses. The purpose of TACS is to externally induce rhythmic brain activity that promotes healthy mental function, particularly when the brain might not be able to produce these rhythms on its own. However, TACS is a relatively new technology, and how it works is still unclear. Whether it can strengthen or revive brain rhythms to change mental function has been a topic of considerable debate in the field of brain stimulation. While some studies find evidence of changes in brain activity and mental function with TACS, others suggest that the currents typically used in people might be too weak to have a direct effect. When faced with conflicting data in the scientific literature, it can be helpful to conduct a type of study called a meta-analysis that quantifies how consistent the evidence is across several studies. A previous meta-analysis conducted in 2016 found promising evidence for the use of TACS in changing mental function. However, the number of studies has more than doubled since then. The design of TACS technologies has also become increasingly sophisticated. We set out to perform a new meta-analysis of studies using TACS to change mental function. To our knowledge, this work is the largest and most comprehensive meta-analysis yet on this topic, consisting of over 100 published studies with a combined total of more than 2,800 human participants. After compiling over 300 measures of mental function across all the studies, we observed consistent and immediate improvement in mental function with TACS. When we examined specific cognitive functions, such as memory and attention, we observed that TACS produced the biggest improvements in executive function, or the ability to adapt in the face of new, surprising, or conflicting information. We also observed improvements in the ability to pay attention and memorize information for both short and long periods of time. Together, These results suggest that tax could particularly improve specific kinds of mental function, at least in the short term. To examine the effectiveness of tax for those particularly vulnerable to changes in mental function, we examined the data from studies that included older adults and people with neuropsychiatric conditions. In both populations, we observed reliable evidence for improvements in cognitive function with tax. Interestingly, we also found that a specialized type of tax that can target two brain regions at the same time and manipulate how they communicate with each other can both enhance or reduce cognitive function. This bidirectional effect on mental function could be particularly useful in the clinic. For example, some psychiatric conditions like depression may involve a reduced ability to process rewards, while others like bipolar disorder may involve a highly active reward processing system. If tax can change mental function in either direction, researchers may be able to develop flexible and targeted designs that cater to specific clinical needs. Developments in the field of tax are bringing researchers closer to being able to safely enhance mental function in a non-invasive way that doesn't require medication. Current statistical evidence across the literature suggests that tax holds promise, and improving its design could help it produce bigger, long-lasting changes in mental function. This article was originally published on The Conversation by Shrey Grover at Boston University. Read the original article here. This emerging brain stimulation technology could boost cognitive function. Copyright provided by Inverse. Related video: 3 mind-body exercises to improve focus in the digital age today. View on Watch. Continue reading. Ad. Visit Inverse. 40 years ago, Apple changed computers forever. Can it happen again? New study finally sheds light on one of the universe's weirdest objects. The crucial lesson, the zone of interest, took from Nope. Ad. Sponsored content. This wrinkle cream sold out at Walmart in 2 days. Governance management platform. Trusted board portal software. Board management software. Trusted board portal software. 7 ways to retire comfortably with $500,000. More. Kirigami electrodes unfold new horizons for brain organoid research. It all started at Pete's. 10 years ago, neuroscientist Sergiu Pasca was just getting his lab up and running. He had not yet published the research for which he would become renowned, a novel technique for growing 3D clusters of human brain cells, often called brain organoids, in petri dishes. Already, Pasca was confident that these organoids and more complex assembloids, which mimic many aspects of the growth and maturation of human brain circuits, would soon let scientists study brain development more precisely than ever before, enabling new insights into the early drivers of conditions like autism or epilepsy. Realizing that potential, however, was a daunting challenge. As we began building neural tissues that were ever more complex, the main limitation was not how we make them, but actually how we're going to precisely probe and manipulate them, said Pasca, now the Kenneth T. Norris, Junior II Professor of Psychiatry and Behavioral Sciences. That's when he ran into chemist Bianchio Cui at a cafe at the James H. Clark Center, which sits between the Stanford School of Medicine and the university's chemistry and biology buildings, and was then the home of the Wu Tsai Neurosciences Institute. Cui is an expert at designing unusual devices and approaches to record the electrical activity of neurons and other cells. They sat down together, and Pasca told Cui about his as-yet unpublished organoid research. That was amazing and quite eye-opening, said Cui, the Job and Gertrude Tamaki professor in the Department of Chemistry. The problem Pasca presented was intriguing: how to record from free-floating clusters of neurons over their months-long development, without catastrophically disrupting their structure and electrical connections. Solving that problem took over a decade of concerted effort, but Cui, Pasca, and their team have finally cracked it. Their approach, inspired by kirigami, an origami-like art form that involves cutting and folding paper to make intricate sculptures, was published in the journal Nature Biotechnology. The study was spearheaded by Wu Tsai neuro-interdisciplinary postdoctoral scholar Xiao Yang and Chui Lab postdoc Chaba Faro. Those years of effort and their ultimate payoff would have been impossible without the impressive array of expertise housed within a half-mile radius at Stanford, Pasca says, the sort of place where a chemist and a physician-scientist, Pasca, trained as a psychiatrist, can meet by chance at a campus cafe and begin a decade-long collaboration, as well as the support of forward-thinking and risk-tolerant organizations like the Wu Tsai Neurosciences Institute. The challenges ahead had been clear from Pasca and Chui's first conversation. Traditional recording techniques are designed to measure brain activity in living animals or in cells cultured on a fixed surface. Brain organoids, however, float freely in fluid and are easily damaged by recording devices. And since organoids are much smaller than an entire brain, even a little damage can be catastrophic. Some scientists record from organoids by slicing them in half and placing the cut edge on an electrically conductive surface, but this approach has downsides, too. In just a few days, they become like a pancake, Chui said. They're no longer free-floating and lose some of their unique properties. Because brain organoids are so different from the tissues neuroscientists have historically worked with, designing approaches for studying and manipulating them demanded a wide diversity of expertise. So Pasca got together several Stanford scientists to found the Stanford Brain Organogenesis Program, which he leads as the Bonnie Waitangsu and Family Endowed Director. Pasca, Chui, and their team attacked the problem of recording from organoids with vigor, even having devices fabricated in Europe when clean rooms at Stanford shut down due to the COVID-19 pandemic. In 2022, Pasca, Chui, and Stanford Brain Organogenesis Program member Jianan Bao, the K. Lee Professor of Chemical Engineering, published a paper reporting on the fruit of their labors, a flexible, mesh-like electrode that could be partly inserted into an organoid. But their quest wasn't over. It did not work well, Chui says. Because the organoids were only affixed to the electrodes on one end, the fluid flowing around them would sometimes tear them entirely free. Their next attempt, however, proved far more successful. Instead of plugging the electrode into the organoid, like a stick poked into a marshmallow at a campfire, the team used a kirigami-inspired approach to fabricate a device that could fully cradle the organoid. Initially, the device looks like an intricate, electrically conductive doily. Once researchers place an organoid at its center, however, it bends and twists into a basket-like shape. The organoid then continues to grow, eventually engulfing the device's supportive strands. For the hammock-like electrode to work, it needed to be flexible enough to allow the organoid to grow naturally and stable enough to stay intact over many months of recording. Balancing those objectives was tricky, so the researchers used computer simulations to aid their iterative design process. The result is extremely effective and extremely delicate. It's easy to tear it, Chui said. You touch it with a pipette, you'll break it. Already, though, Pasca, Chui, and their team have demonstrated that the electrode can record from organoid cells for months, orders of magnitude longer than previous approaches, apparently without disturbing its natural development. This success allowed the team to rigorously answer a question that had previously proved tricky to address: when, exactly, do organoids become electrically active? Because this new device can record from organoids for so long, with no apparent effect on their development, the researchers were able to demonstrate that their organoids started firing action potentials when they were around 100 days old. We've never been able to continuously monitor one neural organoid to see when electrical activity emerges, Pasca said. And that's actually a fundamental question, even for understanding human brain development. The team has their sights set on even more ambitious questions. Pasca's team has recently grown groups of organoids representing different components of brain tissue termed assembloids, that can capture the formation and activity of specific human brain circuits and to study their susceptibility to neurodevelopmental disease. They envision next-generation Kirigami devices that incorporate not only electrodes, but also sensors for substances like oxygen and chemical neurotransmitters, or LEDs that can stimulate cells using optogenetics, a common tool in neuroscience research developed by Stanford Brain Organogenesis Program member Carl Dieseroth, the D.H. Chun Professor of Bioengineering and of Psychiatry. Like any new methodology, these Kirigami electronics will see their fullest realization when taken up by the scientific community, which will be accelerated by the popular hands-on course on neural organoids and assembloids the Brain Organogenesis team hosts each year on the Stanford campus, drawing scientists from around the world. Above all, Pasca said, it's key to recognize that this is a tool designed to advance researchers' ability to understand the mysteries of how the human brain organizes itself early in life, and hopefully develop new approaches to repair defective neural circuits. I think the power of this technology will be in how people use it. More information. Xiao Yang et al., Kirigami Electronics. Transcranial magnetic stimulation. Add. Follow. 9MO. Health topics mentioned in this article. Obsessive-compulsive disorder. Migraine. Smoking. Overview. Transcranial magnetic stimulation, TMS, is a procedure that uses magnetic fields to stimulate nerve cells in the brain to improve symptoms of major depression. It's called a non-invasive procedure because it's done without using surgery or cutting the skin. Approved by the U.S. Food and Drug Administration, FDA, TMS usually is used only when other depression treatments haven't been effective. The FDA also approved TMS for obsessive-compulsive disorder, OCD, migraines, and to help people stop smoking when standard treatments haven't worked well. Research continues into other potential uses for TMS, including epilepsy. When used for depression, OCD, and to stop smoking, this treatment involves delivering repeated magnetic pulses, so it's called repetitive TMS or rTMS. How it works. During an rTMS session for depression, an electromagnetic coil is placed against the scalp of your head. This coil delivers magnetic pulses that stimulate nerve cells in the region of your brain involved in mood control and depression. It's thought to activate regions of the brain that have decreased activity during depression. You also may hear the term deep transcranial magnetic stimulation, or deep TMS. The difference between rTMS and deep TMS has to do with the type of coil used. The deep TMS coil stimulates deeper and wider areas of the brain, compared with rTMS. Deep TMS coils have been FDA approved for OCD and to stop smoking. Although the biology of why TMS works isn't completely understood, the stimulation appears to affect how the brain is working. It seems to ease depression symptoms and improve mood. There are different ways to perform TMS. Methods may change as experts learn more about the most effective ways to perform treatments. Why it's done. Depression is a treatable condition, but for some people, standard treatments aren't effective. Repetitive TMS may be used when standard treatments such as medicines and talk therapy, known as psychotherapy, don't work. TMS is sometimes used to treat OCD, migraines, and to help people stop smoking after other treatments have not been successful. Risks. Repetitive TMS is a non-invasive form of brain stimulation. Unlike vagus nerve stimulation or deep brain stimulation, rTMS does not require surgery or implanting electrodes. And unlike electroconvulsive therapy, ECT, rTMS doesn't cause seizures or memory loss. It also doesn't require the use of anesthesia, which puts people in a sleep-like state. Generally, rTMS is considered safe and well tolerated. However, it can cause some side effects. Common side effects. Side effects are generally mild to moderate and improve shortly after a session. Side effects decrease over time with more sessions. Side effects may include scalp discomfort and pain, headache, tingling, spasms, or twitching of facial muscles, lightheadedness. Your healthcare provider can adjust the level of stimulation to reduce symptoms. Or your provider may recommend that you take a pain medicine available without a prescription before the procedure. In some people who get frequent headaches or migraines, TMS triggers headaches, so treatments might need to be stopped. Uncommon side effects. Serious side effects are rare but may include seizures, emotional highs called mania, particularly in people with bipolar disorder, hearing loss if the ears aren't well protected during treatment. More study is needed to determine whether rTMS may have long-term side effects. How you prepare. Before having rTMS, you may need a physical exam and possibly lab tests or other tests. Mental health evaluation to discuss your depression. These evaluations help make sure that rTMS is a safe option for you. Tell your healthcare provider if you're pregnant or thinking of becoming pregnant. You have metal or implanted medical devices in your body. In some cases, people with metal implants or devices can have rTMS. But due to the strong magnetic field produced during rTMS, it's not recommended for some people who have these devices. Aneurysm clips or coils. Stents. Implanted stimulators. Implanted vagus nerve or deep brain stimulators. Implanted electrical devices, such as pacemakers or medicine pumps. Electrodes for monitoring brain activity. Cochlear implants for hearing. Magnetic implants. Bullet fragments. Other metal devices or objects implanted in their body. You're taking medicines, including prescriptions, medicines available without a prescription, herbal supplements, vitamins or other supplements, and the doses. You have a history of seizures or a family history of epilepsy. You have other mental health conditions, such as issues with alcohol or drugs, bipolar disorder, or psychosis. You have brain damage from illness or injury, such as a brain tumor, stroke, or traumatic brain injury. You have frequent or severe headaches. You have any other medical conditions. You've had treatment with rTMS in the past and whether it was helpful in treating your depression. Before your first appointment. Repetitive TMS isn't invasive, doesn't require anesthesia, and can be performed on an outpatient basis. While you don't need to arrange for someone to drive you home after treatment, you may prefer to be driven home after the first session to get a sense of how you'll feel afterward. Before considering treatment, check with your health insurance company to see whether rTMS is covered. Most policies cover rTMS for depression, but require certain medical necessity conditions to be met. These conditions include trying a minimum number of antidepressants that have not worked. Repetitive TMS for OCD and for stopping smoking is new enough that most insurance policies don't cover its use for these issues. That may change as its use becomes more widespread. What you can expect. Repetitive TMS is usually done in a healthcare provider's office or clinic. It requires a series of treatment sessions to be effective. Generally, sessions are carried out daily, 5 times a week, for 4 to 6 weeks. Your first treatment. Before treatment begins, your healthcare provider will need to identify the best place to put the magnet on your head and the best dose of magnetic energy for you. Your first appointment usually lasts about an hour. During your first appointment, the mapping process is performed. Here's what you can likely expect. You'll be taken to a treatment room, asked to sit in a reclining chair, and given earplugs to wear during the procedure. An electromagnetic coil will be placed against your head and switched off and on repeatedly to produce stimulating pulses. This results in a tapping sensation on your head and a clicking sound, followed by a pause. The coil will be moved around your head and the magnetic energy adjusted to find the location that causes the fingers or hands on the other side of your body to twitch. Once the location is found, a second process of finding the motor threshold occurs. Strong and weak pulses alternate to determine how much energy is needed to move your fingers or thumb during at least half of the attempts. During each treatment, the coil placement and dose are usually done only once. This occurs during the first treatment. For all later treatments, the magnet is moved to the treatment position using measurements from the first treatment. Here's what to expect during each treatment. You'll sit in a comfortable chair, wearing earplugs, with the magnetic coil placed against your head. The treatment location of the coil is different from where the coil makes your fingers or thumb move. When the machine is turned on, you'll feel and hear rapid tapping on your scalp. The pattern will be a few seconds of tapping followed by a pause. The pattern will repeat. You may have scalp discomfort and even some pain during the tapping, but not during the pauses. You'll remain awake and alert during the procedure. Depending on the type of stimulation pattern used, the procedure will last 3.5 minutes or 20 minutes. The latest stimulation type is called intermittent theta burst stimulation and requires only 3.5 minutes. The original RTMS pattern took 37 minutes, but it can be performed in 20 minutes now. After each treatment, you can return to your normal daily activities after your treatment. You might have a headache for a short time afterward. Between treatments, you can usually expect to work and drive. Results. If RTMS works for you, your depression symptoms may improve or go away completely. Symptom relief may take a few weeks of treatment. The effectiveness of RTMS may improve as researchers learn more about techniques, the number of stimulations needed, and the best sites on the brain to stimulate. Ongoing treatment. After completing an RTMS treatment series for depression, standard care, such as medicine and talk therapy, may be recommended as ongoing treatment. It's not yet known if maintenance RTMS sessions will benefit your depression. This involves continuing treatment when you are symptom-free with the hope that it will prevent the return of symptoms. If your depression improves with RTMS for at least 2 to 3 months, and then you have symptoms later, you can repeat RTMS treatment. Most insurance companies will cover repeat treatment. Copyright 2023 Mayo Foundation for Medical Education and Research, MRMER. All rights reserved. In repetitive transcranial magnetic stimulation, RTMS, an electromagnetic coil placed against the scalp creates a magnetic field that stimulates certain areas of the brain. Copyright 2020. Decoding the Brain, a comparative study of human and non-human primates. Add. Follow. Story by Dr. Stacy Livingston, 3MO. The human brain, with its intricate web of connections and functions, has long been a subject of fascination. An international team of scientists has embarked on a mission to map the genetic, cellular, and structural aspects of both human and non-human primate brains, yielding insights that could reshape our understanding of cognition, evolution, and potential treatments for brain disorders. The Brain Initiative's vision. Funded by the NIH's Brain Initiative, the research aims to understand the brain's cellular diversity and its implications in health and disease. The initiative's vast scope might seem daunting, but by breaking it down into tangible tasks, such as the creation of a detailed cell atlas, researchers have made commendable progress. This atlas, when neighboring those of non-human primates, provides a clearer picture of cell types, proportions, and organization. A comparative approach. The decision to compare human brain structures with non-human primates, including chimpanzees, gorillas, macaques, and marmosets, was deliberate. This comparative analysis has revealed evolutionary differences. For instance, subtle shifts in gene expression in humans have led to alterations in neuronal wiring and synaptic function. This comparison shows how we've evolved differently over time. For example, Small changes in our genes have led to alterations to the neuronal wiring, or the way our brain's nerve cells connect, and how they pass messages to each other with synaptic function. These changes have likely enabled our brains to adapt, learn, and evolve more dynamically. Harnessing data for future insights. The data from this research is invaluable. The Brain Cell Atlas alone encompasses over 3,000 types of brain cells. This extensive cataloging offers insights into the variations in brain cell types between humans and non-human primates. Moreover, the research has also revealed how specific brain cell types correlate with particular diseases, laying the groundwork for potential targeted therapies. Furthermore, a study focusing on cell variations in marmosets has drawn connections between the properties of cells in the adult brain and their developmental stages. This suggests that certain cellular properties in adults might trace back to early developmental phases, offering a fresh perspective on brain development across a lifespan. Evolutionary revelations. One of the standout findings from the research is the differences observed in the anatomy and physiology of neurons in the neocortex between humans and mice. The neocortex, responsible for functions like cognition and language, has undergone significant changes in humans, reflecting the complexities of managing intricate brain circuits. Guiding principles in research. Scientific research, especially of this magnitude, is not without challenges. Recognizing and addressing biases, both personal and systemic, is crucial to maintain the integrity of the findings. The BRAIN Initiative, while ambitious, has been methodical in its approach. By segmenting its mission, validating findings, and leveraging successful models, it ensures that the research remains focused and purposeful. Moreover, the tools and methodologies employed, from imaging techniques to genetic sequencing, play a pivotal role in navigating the complexities of brain mapping. The human brain, as a system, requires a holistic understanding. The detailed cellular blueprints provided by the cell atlases are instrumental in this endeavor. The comparative brain cell mapping research has significantly advanced our understanding of the human brain's complexity and its evolutionary trajectory. The potential of this research to revolutionize treatments for brain disorders and enhance our understanding of cognitive functions is immense, promising a brighter future in neuroscience. Decoding the Brain, a comparative study of human and non-human primates, copyright provided by Certainty News. Miracle Hangover Cures, the study reveals a universal pattern of brain wave frequencies across mammalian species. Add. Follow. Story by Science X Staff 6D. Health topics mentioned in this article: epilepsy, schizophrenia, attention deficit hyperactive disorder. Throughout the brain's cortex, neurons are arranged in 6 distinctive layers, which can be readily seen with a microscope. A team of MIT neuroscientists has now found that these layers also show distinct patterns of electrical activity, which are consistent over many brain regions and across several animal species, including humans. The researchers found that in the topmost layers, neuron activity is dominated by rapid oscillations known as gamma waves. In the deeper layers, slower oscillations called alpha and beta waves predominate. The universality of these patterns suggests that these oscillations are likely playing an important role across the brain, the researchers say. The work is published in Nature Neuroscience. When you see something that's consistent and ubiquitous across cortex, it's playing a very fundamental role in what the cortex does, says Earl Miller, the Pickar Professor of Neuroscience, a member of MIT's Pickar Institute for Learning and Memory, and one of the senior authors of the new study. Imbalances in how these oscillations interact with each other may be involved in brain disorders such as attention deficit hyperactivity disorder, the researchers say. Overly synchronous neural activity is known to play a role in epilepsy, and now we suspect that different pathologies of synchrony may contribute to many brain disorders, including disorders of perception, attention, memory, and motor control. In an orchestra, one instrument played out of synchrony with the rest can disrupt the coherence of the entire piece of music, says Robert DeSimone, director of MIT's McGovern Institute for Brain Research and one of the senior authors of the study. Andre Bastos, an assistant professor of psychology at Vanderbilt University, is also a senior author of the paper. The lead authors are MIT research scientist Diego Mendoza Halliday and MIT postdoc Alex Major. Layers of activity. The human brain contains billions of neurons, each of which has its own electrical firing patterns. Together, groups of neurons with similar patterns generate oscillations of electrical activity, or brainwaves, which can have different frequencies. Miller's lab has previously shown that high-frequency gamma rhythms are associated with encoding and retrieving sensory information, while low-frequency beta rhythms act as a control mechanism that determines which information is read out from working memory. His lab has also found that in certain parts of the prefrontal cortex, different brain layers show distinctive patterns of oscillation, faster oscillation at the surface and slower oscillation in the deep layers. One study, led by Bastos when he was a postdoc in Miller's lab, showed that as animals performed working memory tasks, lower frequency rhythms generated in deeper layers regulated the higher frequency gamma rhythms generated in the superficial layers. In addition to working memory, the brain's cortex also is the seat of thought, planning, and high-level processing of emotion and sensory information. Throughout the regions involved in these functions, neurons are arranged in 6 layers, and each layer has its own distinctive combination of cell types and connections with other brain areas. The cortex is organized anatomically into 6 layers, no matter whether you look at mice or humans or any mammalian species, and this pattern is present in all cortical areas within each species, Mendoza Halliday says. Unfortunately, a lot of studies of brain activity have been ignoring those layers because when you record the activity of neurons, it's been difficult to understand where they are in the context of those layers. In the new paper, the researchers wanted to explore whether the layered oscillation pattern they had seen in the prefrontal cortex is more widespread, occurring across different parts of the cortex and across species. Using a combination of data acquired in Miller's lab, DeSimone's lab, and labs from collaborators at Vanderbilt, the Netherlands Institute for Neuroscience, and the University of Western Ontario, the researchers were able to analyze 14 different areas of the cortex from 4 mammalian species. This data included recordings of electrical activity from 3 human patients who had electrodes inserted in the brain as part of a surgical procedure they were undergoing. Recording from individual cortical layers has been difficult in the past. because each layer is less than a millimeter thick, so it's hard to know which layer an electrode is recording from. For this study, electrical activity was recorded using special electrodes that record from all of the layers at once, then feed the data into a new computational algorithm the authors designed, termed FLIP, Frequency-Based Layer Identification Procedure. This algorithm can determine which layer each signal came from. More recent technology allows recording of all layers of cortex simultaneously. This paints a broader perspective of microcircuitry and allowed us to observe this layered pattern, Major says. This work is exciting because it is both informative of a fundamental microcircuit pattern and provides a robust new technique for studying the brain. It doesn't matter if the brain is performing a task or at rest and can be observed in as little as 5 to 10 seconds. Across all species, in each region studied, the researchers found the same layered activity pattern. We did a mass analysis of all the data to see if we could find the same pattern in all areas of the cortex, and voila, it was everywhere. That was a real indication that what had previously been seen in a couple of areas was representing a fundamental mechanism across the cortex, Mendoza-Halliday says. Maintaining balance. The findings support a model that Miller's lab has previously put forth, which proposes that the brain's spatial organization helps it to incorporate new information, which carried by high-frequency oscillations, into existing memories and brain processes, which are maintained by low-frequency oscillations. As information passes from layer to layer, input can be incorporated as needed to help the brain perform particular tasks, such as baking a new cookie recipe or remembering a phone number. The consequence of a laminar separation of these frequencies, as we observed, may be to allow superficial layers to represent external sensory information with faster frequencies, and for deep layers to represent internal cognitive states with slower frequencies, Bostow says. The high-level implication is that the cortex has multiple mechanisms involving both anatomy and oscillations to separate external from internal information. Under this theory, imbalances between high and low-frequency oscillations can lead to either attention deficits such as ADHD, when the higher frequencies dominate and too much sensory information gets in, or delusional disorders such as schizophrenia, when the low-frequency oscillations are too strong and not enough sensory information gets in. The proper balance between the top-down control signals and the bottom-up sensory signals is important for everything the cortex does, Miller says. When the balance goes awry, you get a wide variety of neuropsychiatric disorders. The researchers are now exploring whether measuring these oscillations could help to diagnose these types of disorders. They are also investigating whether rebalancing the oscillations could alter behavior, an approach that could one day be used to treat attention deficits or other neurological disorders, the researchers say. The researchers also hope to work with other labs to characterize the layered oscillation patterns in more detail across different brain regions. Our hope is that with enough of that standardized reporting, we will start to see common patterns of activity across different areas or functions that might reveal a common mechanism for computation that can be used for motor outputs, for vision, for memory and attention, etc., Mendoza-Halliday says. More information. A ubiquitous spectrolaminar motif of local field potential power across the primate cortex. Nature Neuroscience, 2024. DI 10.1038/S41593-023-01554-7. www.nature.com/articles/S41593-023-01554-7. This story is republished courtesy of MIT News, WEB, MIT. edu/news office, a popular site that covers news about MIT research, innovation, and teaching. Provided by Massachusetts Institute of Technology. This story was originally published on Medical Express. Subscribe to our newsletter for the latest SciTech news updates. Credit: Pixabay/CC0 public domain copy. Long-term respiratory illness is here to stay. Have you felt like that winter cold just doesn't end? Well, you're not alone. Many who contracted COVID 19 have suffered from long COVID when the symptoms endure an extended period and can have long term effects. Now, scientists have found that the common cold and flu can also have long versions, which includes symptoms becoming chronic. In addition, we are in the midst of a triple demic with COVID 19, the flu, and RSV all making the rounds. Experts posit that longer term illness is here to stay and more people are likely to be affected in the future. What defines a long illness? A long illness has symptoms lasting well over what is considered to be the average time for the disease. For most respiratory ailments, the threshold is 3 months of symptoms. Some have wrongfully thought they were suffering from a long cold or long flu because of the disremembering that happened during the pandemic. Following the pandemic, when many common viruses weren't circulating widely, some people simply forgot how long symptoms can linger after a standard respiratory illness, usually a period of about 2 weeks, NBC News reported. More severe initial infections are more likely to lead to a long infection, especially when it comes to the flu, according to a study published in the Lancet Infectious Diseases. Conceptualizing these diseases as acute illnesses is really just looking at the tip of the iceberg and eclipses the much higher toll of adverse health outcomes that are happening in the post-acute phase, Dr. Ziad Al-Ali, who led the research, told The Guardian. Yahoo Life explained that long colds and flus can cause symptoms that include a lingering cough, trouble sleeping, memory issues, and trouble with taste and smell, even after a person tests negative for the disease. For some, symptoms can last well past the 3 month mark, and these protracted effects are essentially untreatable. It's really treating symptoms at this point, Dr. Thomas Russo, chief of infectious disease at the University at Buffalo, told Yahoo Life. We're still sorting out what's causing it. That makes treatment difficult. Sometimes long illnesses can produce a secondary illness, like bronchitis or pneumonia. Russo explained, the telltale signs of a bacterial infection on top of influenza, COVID, or a cold is often that a person might start to feel better and suddenly develop a fever, increased shortness of breath, and chest pain. Why is long respiratory illness becoming more prevalent? While experts have not definitively pinpointed a cause, there are a number of theories as to why we are seeing more cases of long respiratory illness. One of the theories, according to NBC News, is that many people are more susceptible to respiratory illnesses this winter because they haven't had a recent infection or vaccination, or that some may have gotten back-to-back infections that they confused with lingering symptoms. There has been a lack of exposure to respiratory diseases over the past 3 years because of masking and social distancing protocols. As such, people are feeling the force of their illnesses more acutely than before, a phenomenon known as immunity debt. People have also been receiving less vaccinations less against the flu, COVID, and RSV. We learned from COVID 19 that infections that are initially thought to cause only acute illnesses can cause chronic disease, Al-Ali told Time. According to the Lancet study, there are 94 health risks associated with COVID and the flu, any of which can develop after someone has been hospitalized for either of the conditions. COVID 19 still has a higher risk of long infection, though. Ongoing research into the long-term effects of COVID 19 and other acute respiratory infections is important because it can help us to get to the root of why some people experience more prolonged symptoms than others. Adrian Martineau, a clinical professor of respiratory infection and immunity at Queen Mary University of London, commented to The Guardian. Ultimately, this could help us to identify the most appropriate form of treatment and care for affected people. Rethinking Consciousness, the ethical debate surrounding AI and animals. Ad. Follow. Opinion by Amir Sheikh 2D. In the current discourse surrounding artificial intelligence, AI, a significant ethical dilemma emerges as comparisons between AI and animal intelligence gain traction. The struggle to define consciousness, especially when viewed through the lens of human experience, raises questions about the ethical implications of such comparisons. The language used in discussing the complexities of AI and animals has the potential to shape public perception and influence the treatment of both entities. This story delves into the nuances of this debate, examining the historical context, ethical concerns, and the impact of language on our understanding of consciousness in AI and animals. Exploring consciousness, bridging the gap between AI and animal minds. The quest to understand consciousness in non-human entities has been a persistent challenge for scientists and philosophers alike. Consciousness, broadly defined as a state of self-awareness, poses difficulties in evaluation due to its subjective nature. In animals, evidence of consciousness is sought through markers such as the ability to feel pain, remember specific events, exhibit self-awareness, and express emotions. Recent discoveries, like the acknowledgement of pain in bumblebees, challenge previous assumptions and highlight the evolving understanding of consciousness in the animal kingdom. A noticeable trend in recent discussions revolves around comparing the complexity of AI to living creatures, with an initial focus on human consciousness. Yet, when AI is deemed distinct from human brains, attention shifts to questions about the cognitive capabilities of animals. This line of inquiry raises ethical concerns, as it inadvertently reinforces the notion that animals and machines share a common category. The language used in such discussions can create a division between human and non-human consciousness, echoing a historical perspective that has long impacted animal rights. Tracing the arc of animal rights. Throughout history, animals were often perceived as intricately designed machines, devoid of consciousness and capable only of responding to pre-programmed rules. This perspective led to the belief that animals could not experience suffering, resulting in limited rights and protections for them. Dedicated research over the years has overturned these assumptions, granting various species rights based on evidence of consciousness. The recent extension of the Animal Sentience Bill in the UK exemplifies the slow evolution of perspectives on animal rights, acknowledging the consciousness of decapod crustaceans and cephalopods. Language holds a powerful influence, especially in realms as delicate as animal rights. The comparison of animals to machines in the past has allowed for the mistreatment of animals by reinforcing the perception of them as unfeeling objects. The potential impact of language on shaping attitudes towards animals cannot be understated. As discussions about AI and animals continue, the careful choice of language becomes crucial to prevent undermining the progress made in understanding and extending empathy towards animals. In contemplating the intersection of AI and animal consciousness, the ethical dimensions become increasingly apparent. The comparison of AI to animals has broader implications for our understanding of consciousness, influencing attitudes towards the rights and protections afforded to non-human entities. As we navigate this intricate terrain, it is imperative to consider the historical context, the evolving perspectives on animal rights, and the influential role of language. The question lingers: can we avoid perpetuating historical biases and ensure that our language reflects the progress made in recognizing and empathizing with the consciousness of animals? Rethinking Consciousness, the ethical debate surrounding AI and animals copyright provided by Cryptopolitan. Related video, raw video, creepy. Deaf boy can now hear after breakthrough gene treatment. Ad. www.nasdaq.com/board_directors/portal trusted board portal software used by 4.5K plus global companies. Ad. www nasdaq.com/board_. Ears. His father's voice, the sounds of passing cars and scissors clipping his hair. An 11-year-old boy is hearing for the first time in his life after receiving a breakthrough gene therapy. The Children's Hospital of Philadelphia, CHOP, which carried out the treatment, a first in the United States, said in a statement Tuesday the milestone represents hope for patients around the world with hearing loss caused by genetic mutations. Sam Dam was born profoundly deaf because of a highly rare abnormality in a single gene. Gene therapy for hearing loss is something that we physicians and scientists in the world of hearing loss have been working toward for over 20 years, and it is finally here, said Surgeon John Germiller, Director of Clinical Research for CHOP's Otolaryngology Division. While the gene therapy we performed in our patient was to correct an abnormality in one very rare gene, these studies may open the door for future use for some of the over 150 other genes that cause childhood hearing loss. In patients like ASAM, a defective gene prevents the production of otoferlin, a protein necessary for the hair cells of the inner ear to be able to convert sound vibrations into chemical signals that are sent to the brain. Otoferlin gene defects are highly rare, accounting for 1 to 8% of hearing loss present from birth. On October 4, 2023, he underwent a surgical procedure that involved partly lifting his eardrum and then injecting a harmless virus, which had been modified to transport working copies of the otoferlin gene, into the internal fluid of his cochlea. As a result, the hair cells began making the missing protein and functioning properly. Almost 4 months since receiving the treatment in one ear, Isam's hearing has improved to the point he only has mild to moderate hearing loss, and he is literally hearing sound for the first time in his life, said the statement. The New York Times reported that despite being able to hear, Sam, who was born in Morocco and later moved to Spain, may never learn to talk, as the brain's window for acquiring speech closes around the age of 5. The U.S. Food and Drug Administration, which greenlighted the study, wanted to start the research on older children first, for safety reasons. The trial, sponsored by Akouos, Inc., a wholly owned subsidiary of Eli Lilly and Company, is one of several underway or about to start in the United States, Europe, and China, where a handful of other children have already been reportedly cured. As more patients at different ages are treated with this gene therapy, researchers will learn more about the degree to which hearing is improved and whether that level of hearing can be sustained over many years, Germiller said. I/Neuro. Sam Dam, third from left, was born profoundly deaf because of a highly rare abnormality in a single gene. Copyright handout. Related video. Baby hears her father's voice clearly for the very first time and gets the fright of her life. Daily Motion. FDA to require new warning of secondary cancer risk for CAR-T cancer therapies. The Food and Drug Administration, FDA, is requiring a boxed warning on all existing CAR-T cancer treatments following a review of reports that the therapies themselves can increase the risk for some secondary cancers. In letters to the manufacturers of all 6 currently available CAR-T therapies dated January 19, the FDA said the prescribing information will need to include so-called black box warnings, the most serious type for any medication. CAR-T is a gene therapy that works by extracting white blood cells known as T cells from a patient, re-engineering them in a lab to attack cancer cells, and then infusing them back into the body. The treatments impacted include Abecma and Breyanzi from Bristol Myers Squibb, Carvykti from Johnson & Johnson, J&J, and Legend Biotech, Kymriah from Novartis and Tecartus, and Yescarta from Gilead. The required warning label comes less than 2 months after the FDA made public an investigation into reports of T-cell malignancies, types of cancer in blood cells, including lymphoma and leukemia in people who received treatment with CAR-T therapies. The FDA at the time said it determined that the risk was serious and applicable to all currently approved CAR-T products. Some of the disorders resulted in hospitalization and death. The agency is allowing the therapies to stay on the market, a sign their benefits continue to outweigh the risks. The companies have 30 days to submit proposed changes to the label. Kymriah, the first CAR-T therapy to get FDA approval, has been on the market since 2017 to treat leukemia. The FDA's initial approvals of CAR-T products included requirements to conduct 15-year-long follow-up safety studies to assess the long-term safety and the risk of secondary malignancies occurring after treatment. Novartis and J&J separately said they would work with the FDA to update the prescribing information for Kymriah and Carvykti. Novartis added it has not found sufficient evidence to support the causal relationship between Kymriah and secondary T-cell malignancies to date and remains confident in the favorable benefit/risk profile of Kymriah. A spokesperson for Gilead similarly said the company will work with the FDA on next steps, but is not aware of evidence to date establishing that treatment with Yescarta or Tecartus has a causal role in the development of malignancies of T-cell origin. For the latest news, weather, sports, and streaming video, head to The Hill. Plant cell discovery can potentially reverse aging by hacking aging process. Scientists in the United States have found a plant protein that they believe can hack the aging process. After research at the University of California, Riverside, the Golgi apparatus, which is an organelle, can play a role in reversing aspects of aging in humans. Organelles are found in both plant and human cells, and their role is crucial to the healthy functioning of these species. They process essential proteins and lipids before secreting them or moving them onto other cells. They are named after Camillo Golgi, who was an Italian scientist who was researching the nervous system in 1898. While studying plant stressors, the team at UCR discovered an element of organelles that could potentially preserve a cell's longevity, and this could have implications on aging in humans. For us, this finding is a big deal, said Katie Dehesh, a co-author of the study and a professor of molecular biochemistry at the Californian University. For the first time, we have defined the profound importance of an organelle in the cell that was not previously implicated in the process of aging. Their research initially focused on how Thale-Crest plant cells reacted to external stress, such as infection and poor light. However, the research team soon found that the Golgi apparatus, thanks to a protein it contains, protected the cell against and helped it survive against external factors. Proteins known as conserved oligometric Golgi, COG, help the organelle attach carbohydrates or sugars to other proteins and lipids. These proteins are then transported to other cells. The process is called glycosylation, and this helps with the healthy functioning of cells and other crucial biological functions. Golgi are like the post office of the cell, according to Heesung Choi, who is the study lead author and a researcher in the Botany and Plant Sciences department at UCR. They package and send out proteins and lipids to where they're needed. A damaged Golgi can create confusion and trouble in the cell's activities, affecting how the cell works and stays healthy. The protein ensures the movement of small envelopes that circulate molecules around the cell, which can be crucial in the process of protecting the cell from externally arising issues. In the study, the researchers modified plants so that they did not contain the protein. Initially, they grew normally, but they soon wilted when they were deprived of sunlight and were unable to convert it into sustenance via photosynthesis. These plants were shown to decline 3 times faster than those that were not modified and contained the COG protein. In the dark, the COG mutants showed signs of aging that typically appear in wild, unmodified plants around day 9, said Choi. But in the mutants, These signs manifested in just 3 days. The research team found that when they enabled the modified plants to create the COG protein again, they were revived to become normal plants once again. It was like nothing happened to them once we reversed the mutation, according to Dahesh. These responses highlight the critical importance of the COG protein and normal Golgi function in stress management, Choi said. While the research applied to plants, human cells also have a Golgi apparatus, post-office, inside them. The human COG complex, which is comprised of 8 protein subunits, can impact crucial biological roles such as glycosylation and protein sorting when they don't work as they should. This malfunctioning has been shown as a factor in the growth of cancer cells and other diseases, showing how crucial the protein is to a person enjoying good health. The research now wants to study the disruption to this pathway in human cells and examine any potential relationship between human COG proteins and the process of aging and stress. There is the potential that this research could lead to therapies that can strengthen cells and shield them from the external issues that can lead to premature aging. Not only does our research advance our knowledge about how plants age, but it could also provide crucial clues about aging in humans, said Dahesh. When the COG protein complex doesn't work properly, it might make our cells age faster, just like what we saw in plants when they lacked light. This breakthrough could have far-reaching implications for the study of aging and age-related diseases. For the latest significantly restricting movement is most effective in stopping disease spread, SARS-CoV-2 data show. Researchers from La Trobe University in Melbourne, Takunenga Ki Purahiro Massey University and Waipapa Taumata Rao, University of Auckland in New Zealand, studied the dynamics of infectious disease transmission in an interconnected world for their paper, High Connectivity and Human Movement Limits the Impact of Travel Time on Infectious Disease Transmission. The study, published in the Journal of the Royal Society Interface, underscores the difficulty of controlling an emerging infection in highly connected communities. The researchers used real-world data focused on the spread of SARS-CoV-2 during the COVID-19 pandemic, with the aim of better understanding how such a disease spreads from person to person in a highly connected global environment. Before vaccination, interventions would often rely on changes in human mobility to curb or eliminate the viral spread of disease. The research team used a population model based on human movement data from 340 cities in China. This model effectively replicates the early trajectory of COVID-19 cases, providing valuable insights into the spread of the virus. Algorithms were used to identify properties influencing the spread between cities. The findings showed that travel time between cities, calculated using the maximum allowed speed for a particular road and distances between through-road networks, was the most crucial factor, followed closely by the numbers of people moving between cities. The findings revealed that minor restrictions in movement aren't enough to significantly impact spread between cities, and only substantial reductions in human movement between cities can significantly slow infection spread times. The authors found these insights offer a nuanced understanding of how infectious diseases spread in a globally connected world and can help to guide future public health interventions and policy decisions. Lead investigator Dr. Riju John from Waipapa Taumata Rao, University of Auckland, says the research provides a sophisticated understanding of the relationship between human movement, connectivity, and travel time in the context of infectious disease transmission. These insights are vital for developing effective strategies to manage and mitigate the impact of pandemics on a global scale, he added. Massey's Percival Carmine Chair in Epidemiology and Public Health, Professor David Heyman, says the research contributes to the ongoing discourse surrounding infectious diseases. It also lays the groundwork for more effective and targeted interventions, emphasizing the need for nuanced epidemiological models that incorporate mobility dynamics, Professor Heyman said. Senior investigator from La Trobe University, Associate Professor Mathematics and Statistics Joel Miller, says the collaborative effort marks a significant advancement in our collective understanding of infectious disease transmission in our interconnected world. This work underscores the difficulty of controlling an emerging infection in highly connected communities, Associate Professor Miller said. Unfortunately, minor reductions in travel are not effective. A pathogen will quickly find alternative routes between communities. More information. Riju Sam John et al. High connectivity and human movement limits the impact. Coming to grips with the implications of quantum mechanics. The question is no longer whether quantum theory is correct, but what it means. For almost a century, physicists have wondered whether the most counterintuitive predictions of quantum mechanics, QM, could actually be true. Only in recent years has the technology necessary for answering this question become accessible, enabling a string of experimental results, including startling ones reported in 2007 and 2010, and culminating now with a remarkable test reported in May that show that key predictions of QM are indeed correct. Taken together, these experiments indicate that the everyday world we perceive does not exist until observed, which in turn suggests, as we shall argue in this essay, a primary role for mind and nature. It is thus high time the scientific community at large, not only those involved in foundations of QM, faced up to the counterintuitive implications of QM's most controversial predictions. Over the years, we have written extensively about why QM seems to imply that the world is essentially mental, e.g. 1990, 1993, 1999, 2001, 2007, 2017a, 2017b. We are often misinterpreted and misrepresented as espousing solipsism or some form of quantum mysticism, so let us be clear. Our argument for a mental world does not entail or imply that the world is merely one's own personal hallucination or act of imagination. Our view is entirely naturalistic. The mind that underlies the world is a transpersonal mind behaving according to natural laws. It comprises but far transcends any individual psyche. The claim is thus that the dynamics of all inanimate matter in the universe correspond to transpersonal mentation, just as an individual's brain activity, which is also made of matter, corresponds to personal mentation. This notion eliminates arbitrary discontinuities and provides the missing inner essence of the physical world. All matter, not only that in living brains, is the outer appearance of inner experience. Different configurations of matter reflecting different patterns or modes of mental activity. According to QM, the world exists only as a cloud of simultaneous, overlapping possibilities, technically called a superposition, until an observation brings one of these possibilities into focus in the form of definite objects and events. This transition is technically called a measurement. One of the keys to our argument for a mental world is the contention that only conscious observers can perform measurements. Some criticize this contention by claiming that inanimate objects, such as detectors, can also perform measurements, in the sense described above. The problem is that the partitioning of the world into discrete inanimate objects is merely nominal. Is a rock integral to the mountain it helps constitute? If so, does it become a separate object merely by virtue of its getting detached from the mountain? And if so, does it then perform a measurement each time it comes back in contact with the mountain as it bounces down the slope? Brief contemplation of these questions shows that the boundaries of a detector are arbitrary. The inanimate world is a single physical system governed by QM. Indeed, as first argued by John von Neumann and re-articulated in the work of One of Us. When two inanimate objects interact, they simply become quantum mechanically entangled with one another, that is, they become united in such a way that the behavior of one becomes inextricably linked to the behavior of the other. But no actual measurement is performed. Let us be more specific. In the well-known double-slit experiment, electrons are shot through two tiny slits. When they are observed at the slits, the electrons behave as definite particles. When observed only after they've passed through slits, the electrons behave as clouds of possibilities. In 1998, researchers at the Weizmann Institute in Israel showed that, when detectors are placed at the slits, the electrons behave as definite particles. At first sight, this may seem to indicate that measurement does not require a conscious observer. However, the output of the detectors only becomes known when it is consciously observed by a person. The hypothesis of a measurement before this conscious observation lacks compelling theoretical or empirical grounding. After all, as discussed above, QM offers no reason why the whole system, electrons, slits, and detectors combined, wouldn't be in an entangled superposition before someone looks at the detector's output. As such, a conscious observer may be indispensable, an idea further elaborated by one of us with regard to so-called delayed choice quantum eraser experiments. The bottom line is that we cannot know that detectors actually perform measurements, for we cannot abstract ourselves out of our knowledge. Recall Max Planck's position: I regard consciousness as fundamental. I regard matter as derivative from consciousness. We cannot get behind consciousness. Emphasis added. Some claim that the modern notion of decoherence rules out consciousness as the agency of measurement. According to this claim, when a quantum system in a superposition state is probed, information about the overlapping possibilities in the superposition leaks out and becomes dispersed in the surrounding environment. This allegedly explains in a fairly mechanical manner why the superposition becomes indiscernible after measurement. The problem, however, is that decoherence cannot explain how the state of the surrounding environment becomes definite to begin with, so it doesn't solve the measurement problem or rule out the role of consciousness. Indeed, as Warcek Zurek, one of the fathers of decoherence, admitted, an exhaustive answer to the question of why we perceive a definite world would undoubtedly have to involve a model of consciousness, since what we are really asking concerns our observers' impression that we are conscious of just one of the alternatives. As a matter of fact, peculiar statistical characteristics of the behavior of entangled quantum systems, namely, their experimentally confirmed violation of so-called Bell's and Leggett's inequalities, seem to rule out everything but consciousness as the agency of measurement. Some then claim that entanglement is observed only in microscopic systems and, therefore, its peculiarities are allegedly irrelevant to the world of tables and chairs. But such a claim is untrue, as several recent studies, e.g. 2009, 2011, and 2015, have demonstrated entanglement for much larger systems. Last year, a paper reported entanglement even for massive objects. Moreover, quantum superposition has been observed in systems as varied as small metal paddles and living tissue. Clearly, the laws of QM apply at all scales and substrates. What preserves a superposition is merely how well the quantum system, whatever its size, is isolated from the world of tables and chairs known to us through direct conscious apprehension. That a superposition does not survive exposure to this world suggests, if anything, a role for consciousness in the emergence of a definite physical reality. Now that the most philosophically controversial predictions of QM have, finally, been experimentally confirmed without remaining loopholes, there are no excuses left for those who want to avoid confronting the implications of QM. Lest we continue to live according to a view of reality now known to be false, we must shift the cultural dialogue towards coming to grips with what nature is repeatedly telling us about herself. Research team observes the processes of memory and cognition in real time. Add. Follow. Story by Science X Staff 5D. The human brain contains approximately 86 billion neurons and 600 trillion synapses that exchange signals between the neurons to help us control the various functions of the brain, including cognition, emotion, and memory. Interestingly, the number of synapses decreases with age or as a result of diseases like Alzheimer's, and research on synapses thus attracts a lot of attention. However, limitations have existed in observing the dynamics of synapse structures in real time. On January 9th, a joint research team led by Professor Won-Do Hyo from the KSD Department of Biological Sciences, Professor Hyungbae Kwon from the Johns Hopkins School of Medicine, and Professor Senkyu Lee from the Institute for Basic Science, IBS, revealed that they had developed the world's first technique to allow real-time observation of synapse formation, extinction, and alterations. Professor Hyo's team conjugated dimerization-dependent fluorescent proteins, DDFP, to synapses in order to observe the process by which synapses create connections between neurons in real time. The team named this technique Synapshot by combining the words synapse and snapshot, and successfully tracked and observed the live formation and extinction processes of synapses as well as their dynamic changes. Through a joint research project, the teams led by Professor Hyo and Professor Senkyu Lee at IBS together designed a synapshot with green and red fluorescence and were able to easily distinguish the synapse connecting two different neurons. Additionally, by combining an optogenetic technique that can control the function of a molecule using light, the team was able to observe the changes in the synapses while simultaneously inducing certain functions of the neurons using light. Through more joint research with the team led by Professor Hyungbae Kwon at the Johns Hopkins School of Medicine, Professor Hyo's team induced several situations on live mice, including visual discrimination training, exercise, and anesthesia, and used Synapshot to observe the changes in the synapses during each situation in real time. The observations revealed that each synapse could change fairly quickly and dynamically. This was the first ever case in which the changes in synapses were observed in a live mammal. Professor Hyo said, "Our group developed Synapshot through collaboration with domestic and international research teams and have opened up the possibility for first-hand live observations of the quick and dynamic changes of synapses, which was previously difficult to do. We expect this technique to revolutionize research methodology in the neurological field and play an important role in brightening the future of brain science." This research, conducted by co-first authors Seungkyu Sun, PhD candidate, Jin-Soo Lee, PhD candidate, and Dr. Kong Hoon-young from Johns Hopkins, was published in the online edition of Nature Methods on January 8 under the title, Real-Time Visualization of Structural Dynamics of Synapses in Live Cells in Vivo. More information: Seungkyu Sun et al., Real-Time Visualization of Structural Dynamics of Synapses in Live Cells in Vivo, Nature Methods, 2024. DI 10.1038/S41592-023-02122-4. Provided by the Korea Advanced Institute of Science and Technology, KAIST. This story was originally published on Medical Express. Subscribe to our newsletter for the latest SciTech news updates. To observe dynamically changing synapses, dimerization-dependent fluorescent protein, DDFP, was expressed to observe fluorescent signals upon synapse formation as DDFP enables fluorescence detection through reversible binding to pre- and postsynaptic terminals. Credit: KAIST Optogenetics and RNA Therapeutics Lab. Copyright provided by Medical Express. Microscopic photos observed through changes of the fluorescence of the synapse sensor, Synapshot, by cultivating the neurons of an experimental rat and expressing the Synapshot. The changes in the synapse that is created when the pre- and postsynaptic terminals come into contact and the synapse that disappears after a certain period of time are measured by the fluorescence of the Synapshot. Credit: KAIST Optogenetics and RNA Therapeutics Lab. Copyright provided by Medical Express. Simultaneous use of green synapshot and red synapshot to distinguish and observe synapses with one post-terminal and different pre-terminals. Credit: KAIST Optogenetics and RNA Therapeutics Lab. Copyright provided by Medical Express. Dimer-dependent fluorescent protein, DDFP, exists as a green fluorescent protein as well as a red fluorescent protein and can be applied together with blue light-activated optogenetic technology. After activating tropomyosin receptor kinase B, TRKB, by blue light using optogenetic technology, the strengthening of synaptic connections through signals of brain-derived neurotrophic factor is observed using red synapshot. Credit: KAIST Optogenetics and RNA Therapeutics Lab. Copyright provided by Medical Express. Micrographs showing real-time changing synapses in the visual cortex of mice trained through visual training using in vivo imaging techniques, such as two-photon microscopy as well as at the cellular level. Credit: KAIST Optogenetics and RNA Therapeutics Lab. Copyright provided by Medical Express. Related video: research suggests cognitive ability associated with more controlled emotional response. Money Talks News. View on watch. Continue reading. OpenAI GPT 4.5 in days will go beyond human experts and more AI predictions for 2024. 2024 and every year going forward will have more and better AI. This intelligence explosion is attracting funding and resources exponentially, and its revenue will go exponential. AI expert Dr. Alan Thompson expects OpenAI to release GPT 4.5 in days, and it will exceed human experts in most intelligence tests. A survey of thousands of AI experts does not expect AI to be capable of surpassing human in all tasks until about 2047, 13 years earlier than last year's survey. And they do not expect AI to replace all human jobs until 2100 to 2150. However, most of them expect AI to accelerate technological development by 10 times within 5 years. How will technological advancement been rapidly accelerated from every month being a year of progress without complete world-altering effects? An NVIDIA expert, Jin Fan, forecasts synthetic data becoming very important. Synthetic data will be a significant research path in 2024. Many old techniques that were meant for board games and image generation are now revived with new meanings for LLMs. This paper's idea is simple: self-play plus GN. Intuitively, an LLM at iteration, T plus 1, plays an adversarial game against its older self at T. T plus 1 tries to tell a model's output from real humans' output, while T tries to fool its opponent by generating text that looks as real as possible. Then the T plus 1 parameters are copied to T, and the self-improvement loop continues. Most importantly, this method does not require human preference data. A supervised fine-tuning (SFT) dataset is enough to kickstart the self-play game. I think this method is similar to GALE in spirit. GALE stands for Generative Adversarial Imitation Learning, which outperforms simple behavior cloning for control tasks like robotics. It also runs this adversarial game that drives self-improvement, but with a physics simulator in the loop. Brian Wang. Brian Wang is a futurist thought leader and a popular science blogger with 1 million readers per month. His blog nextbigfuture.com is ranked #1 science news blog. It covers many disruptive technology and trends including space, robotics, artificial intelligence, medicine, anti-aging biotechnology, and nanotechnology. Known for identifying cutting-edge technologies, he is currently a co-founder of a startup and fundraiser for high-potential early-stage companies. He is the head of research for allocations for deep technology investments and an angel investor at Space Angels. A frequent speaker at corporations, he has been a TEDx speaker, a Singularity University speaker, and guest at numerous interviews for radio and podcasts. He's open to public speaking and advising engagements. Breakthroughs in Artificial Intelligence Research. Add. Follow. Story by John Palmer, 1D. Artificial intelligence, AI, continues to be at the forefront of technological advancements, with researchers and scientists pushing the boundaries of what's possible. Reinforcement learning, achieving superhuman performance. One of the most notable breakthroughs in AI research is the remarkable progress in reinforcement learning algorithms. These algorithms, inspired by behavioral psychology, enable machines to learn and make decisions by interacting with their environment. Autonomous agents mastering complex games. One of the most impressive demonstrations of reinforcement learning is in the realm of gaming. AI agents, using algorithms like AlphaZero and OpenAI's GPT-3, have achieved superhuman performance in games like chess, Go, and even video games like Dota 2. These AI systems have surpassed the capabilities of human players, showcasing the potential of AI in solving complex problems. Real-world applications. Reinforcement learning is not limited to games. It has found applications in robotics, autonomous vehicles, and finance. Robots can learn to perform tasks in unstructured environments, self-driving cars can navigate safely, and trading algorithms can make better investment decisions, all thanks to advancements in reinforcement learning. Natural language processing, understanding human language. Natural language processing, NLP, has seen significant advancements, enabling machines to understand and generate human language. Multilingual models. Multilingual models like BERT and GPT-3 have the ability to understand and generate text in multiple languages. This has profound implications for global communication, breaking down language barriers and facilitating cross-cultural interactions. Contextual understanding. Recent developments in NLP have improved contextual understanding. Machines can now grasp the subtleties of language, making them better at tasks like sentiment analysis, content summarization, and language translation. This has broad applications in content generation, customer support, and data analysis. AI in healthcare. NLP is making waves in healthcare by helping medical professionals analyze vast amounts of patient data and research papers. AI-powered tools can extract valuable insights, aiding in diagnosis and treatment planning. This has the potential to improve healthcare outcomes and save lives. Computer vision, seeing the world through AI eyes. Computer vision, the field of teaching machines to interpret visual information, has witnessed significant advancements. Object recognition. AI systems can now recognize and classify objects in images and videos with high accuracy. This technology is essential in various sectors, from security surveillance to autonomous drones. Facial recognition. Facial recognition algorithms have become more sophisticated, enabling applications in identity verification, security, and personalized marketing. However, ethical concerns regarding privacy and bias must be addressed as this technology matures. Healthcare and autonomous vehicles. Computer vision plays a crucial role in the healthcare industry by assisting in medical image analysis and diagnostics. Additionally, autonomous vehicles rely on computer vision to navigate roads safely, making transportation more efficient and reducing accidents. The field of artificial intelligence continues to make astounding progress in various domains. Reinforcement learning is pushing the boundaries of problem solving, NLP is revolutionizing human-machine interaction, and computer vision is transforming how machines perceive the world. These breakthroughs have profound implications for industries such as healthcare, finance, gaming, and more. However, as AI technology becomes more integrated into our lives, it also raises important ethical and societal questions that must be addressed. As researchers and scientists continue to innovate, we can anticipate even more remarkable advancements on the horizon, shaping a future where AI plays an increasingly integral role in our daily lives. The journey of artificial intelligence is far from over, and the possibilities are limitless. Breakthroughs in artificial intelligence research copyright provided by Cryptopolitan. Related video. Artificial intelligence technology identifies counterfeit drugs, Fox Business. View on watch. Continue reading. Ad. Visit Cryptopolitan. Google Chrome enhances AI features to compete with Microsoft Edge. The four arenas where 2024 will reshape our world. Borderless Capital leads $1.5 million seed round for Synonym Finance to deliver robust. Researchers design new open-source technology for interfacing with living neurons. Ad. Follow. Story by Shel Lawson, 17H. Neurons intricately communicate and respond to stimuli within a vast network, orchestrating essential functions from basic bodily processes to complex thoughts. Traditional neuroscience methods, relying on in vivo electrophysiology within a living organism, often have difficulty addressing the complexity of the brain as a whole. An alternative approach involves extracting cells from the organism and conducting studies on a culture dish instead, in vitro, providing researchers with enhanced control and precision in measuring neural processes. In a new study featured in Advanced Science, researchers unveil a cost-effective, open-source in vitro system for interfacing with neurons, offering a more accessible avenue for researchers interested in neural interactions. The study is part of a larger project called Mind in Vitro, which explores how neurons interact with each other, not only to better understand the functions underlying complex systems like the brain, but also for the goal of eventually using in vitro neural networks for computation. To accomplish such a goal, the MIVI project encompasses a highly interdisciplinary group of researchers, including those from computer science, engineering, neurobiology, physiology, and more. The goal of the MIVI project is to ultimately use neurons for computation, explained G. Andrew Doe, a graduate student in the Gotsola lab who helped lead the project. This would allow for a system that is dynamic and constantly evolving, unlike traditional computing systems, and is actually more energy efficient as well. The new MIVI study, led by Mattia Gotsola, M-Cells, an associate professor of mechanical science and engineering, Xiaotian Zhang, a postdoctoral researcher in Gotsola's lab, and Doe, describes an innovative approach to measuring neuron activity using microelectrode array (MEA) technology. While there are commercial systems that utilize similar technology, they are often expensive and marketed towards specific experimental approaches. The problem with the current technology for interfacing with neurons is that it's mainly commercial systems, which are more standardized for specific testing conditions, generally within biochemistry or traditional neuroscience, said Zhang. Since we're interested in engineering computing paradigms out of living cells, our motivation for this was to move away from the more standardized commercial systems and build something ourselves that we would have full control over. In the new MIVI apparatus, cells are placed onto a plate that contains MEAs, allowing the technology to interface with the neural substrates. The electrodes detect voltage from the neurons, and that voltage is amplified and sent to a computer, which then processes the data. Compared to the standard 60-electrode commercial system, the MIVI system boasts over 500 electrodes, which allows for more data to be collected at once. The system also incorporates improved and/or novel features compared to commercial systems, such as portability, bidirectional communication with neurons, the ability to image neurons during recordings, and the ability to test multiple types of input, electric and optical, and multiple cell populations at a time. Despite these enhancements, the cost to build the MIVI system is shockingly 10 times cheaper than the cost of a commercial system and completely customizable. In order to further the dissemination of the system, the researchers have made their hardware and software models for the MIVI system open source and freely available online. Having our software and hardware be open source was really important to us, said Zhang. We wanted to bring the cost down to make this technology available to others and have it be more flexible and versatile so that researchers can really customize their experimental settings for whatever they need. Our system is not for only one lab or one group of people interested in one specific thing, it's for everyone. We designed our system to be very easy to make and comprised of inexpensive parts, which will allow a lot of labs that can't afford the commercial system to have their own system, said Doe. Even though the original design is for computational studies, we've made the structure easy to redesign and expand upon, so we're pretty sure this will satisfy researchers no matter the kind of studies they want to do. The researchers say other labs are already showing interest in the new technology for their own experiments. The MeV team plans to keep improving the design to make the hardware more automated and accessible, as well as explore ways to quantify and expand the technology's performance. Links for the open-source hardware designs and software can be found at https://gotsolalab, github.io/mev-oh, and https://mev-os, readthdocs.io respectively. Microsoft hires energy mavericks in quest for nuclear-powered data centers. Add. Follow. Story by Dan Robinson, 23H. Industry vets specialize in the development of small modular reactors. Microsoft has hired a director of nuclear technologies to oversee a program to develop small-scale atomic reactors to power data centers as an alternative to fossil fuels. Archana Archie Manoran has been tapped for the role. She spent more than 15 years in the energy industry and was former director of nuclear strategy and programs at Ultra Safe Nuclear Corporation, USENC, a company developing micro-modular reactor, MMR, technologies. Manoran posted on her LinkedIn page that she was delighted to have started at Microsoft as the head honcho of nuclear technologies. Microsoft is proud to be part of the global effort to decarbonize the electricity sector and empower every person and every organization on the planet to achieve more. I am eager to contribute to this vision, she said. At USENC, Manoran oversaw long-range nuclear strategy as well as key stakeholder partnerships for the company's MMR deployments. However, she was also responsible for USENC's Department of Energy, DOE, loan program application efforts, which will likely have been valuable experience as far as Microsoft was concerned. The move follows Microsoft's appointment of Erin Henderson last week as Director of Nuclear Development Acceleration. Another energy industry veteran, she joins Microsoft from Tennessee Valley Authority Electricity Company, where her role was GM of Transmission Projects. As we reported last year, Microsoft was seeking senior staff to bolster its nuclear ambitions, with one job ad posted for a principal program manager for nuclear technology to lead project initiatives for all aspects of energy infrastructure. This includes liaising with engineering and design teams to ensure technical feasibility and optimal integration of SMR and microreactor systems. Microsoft, along with others involved in the data center industry, is looking into nuclear power for two reasons: the need to move away from fossil fuels to cut carbon dioxide emissions, plus the expanding energy consumption of data centers as more powerful hardware is called for to meet the demands of AI processing. Redmond's interest in MMRs or SMRs, small modular reactors, centers on the fact they are small, up to 300 megawatts in output, and modular, and can theoretically be prefabricated elsewhere, then shipped to the data center site and installed, which should make them more affordable to build than the large-scale fission reactors that power cities or entire regions. However, this is a long-term project, as the Register explained last year. One analyst told us at the time that the most optimistic deployment of an SMR here in the United States is by 2030, and deployment to power data centers is likely to be between 10 and 15 years away. Microsoft is clearly playing a long game. Last year, it signed a power purchase agreement with Helion Energy, a company based in Washington state with the ambitious aim of building the world's first operational nuclear fusion power plant. This is optimistically forecast by the company to start producing electricity by 2028, but skeptics have poured cold water on this, pointing out it would be a short time frame to go from blueprint to grid power with an established design of nuclear fission power plant, let alone an entirely unproven tech. Registered. Microsoft hires energy mavericks in quest for nuclear-powered data centers. Copyright provided by the Register. Related video. Nuclear fusion reactor for cleaner energy. Daily Motion. Continue reading. Ad. Visit the Register. Microsoft admits issues with Windows 10 patch almost 2 months after release. XML orders boom, but export restrictions could hamper growth. €2 billion SAP restructure program will affect 8,000 roles. Ad. Sponsored content. Easily manage board meetings. User-friendly board portal. Michigan launches new policy for cars used less than multi-generational toxicant exposures show cumulative, inherited health effects. While exposure to a single substance like DDT has been shown to create inherited disease susceptibility, a recent study in animals found exposure to multiple different toxicants across generations can amplify those health problems. In the study, published in the journal Environmental Epigenetics, an initial generation of pregnant rats was exposed to a common fungicide, then their progeny to jet fuel, and the following generation to DDT. When those rats were then bred out to a fifth unexposed generation, the incidence of obesity as well as kidney and prostate diseases in those animals were compounded, rising by as much as 70%. Researchers also found that their epigenetics, molecular processes independent of DNA that influence gene expression, were also greatly altered. We looked at multiple generation exposures because these types of things are going on routinely, and previous research has only looked at single exposures, said Michael Skinner, a WSU biology professor and the study's corresponding author. We found that if multiple generations get different exposures, then eventually there's an amplification or compounded effect on some diseases. The study did show that for other diseases, those associated with the ovaries and the testes, the incidence rose in the first generation of progeny but appeared to plateau with the additional generational exposures. Skinner and his colleagues also conducted epigenetic analysis of each generation of the animals, finding that the toxicant exposures shifted their entire epigenetics dramatically. Along with changes to genes themselves, inherited epigenetics, which influence gene expression, are considered to have a significant impact on evolution. When we made the comparisons with different generations, we did not find a lot of overlap in epigenetics. In other words, every time each generation had a new exposure, it appeared to reprogram the whole epigenome, Skinner said. While the study does not exactly mimic what may have happened to human generations, people in the US have potentially been exposed to these particular toxicants at different times. The authors note a likely sequential exposure over human generations might involve an exposure to DDT, which was widely used in the 1950s, then plastics in the 1970s, followed by many modern herbicides still in use today. The study provides evidence that multiple toxicant exposures of past human generations likely had a compounded impact on grandchildren and great-grandchildren. Knowing about these possible impacts can help people and doctors address potential diseases before they develop, Skinner said. Researchers, including Skinner's lab at WSU, have been working to identify epigenetic biomarkers for inherited health conditions in humans, including for obesity, autism, and preterm birth. We need to know to what degree our past generations' activities that have affected us, Skinner said. We cannot necessarily fix this problem, but it's important to know that it happens so that we can try to better treat potential health problems through preventative medicine. More information. Eric E. Nielsen et al. Multiple generation distinct toxicant exposures induce epigenetic transgenerational inheritance of enhanced pathology and obesity. Environmental Epigenetics, 2023. DI 10.1093/EAP/DVAD006. Provided by Washington State University. These chemicals, found almost everywhere, can reduce fertility by 40%, study finds. Ad. Sponsored collections LED high bay lights that will brighten up your workspace. Ad. Dell Apex Cloud Services, Multi-Cloud Control. Follow. Story by Megan Holohan, 10MO. Health topics mentioned in this article. Infertility. Cancer. Kidney stone. Many couples with infertility wonder why they struggle to become pregnant. A new study reveals one possible cause after it found that women with PFAS, also called forever chemicals, in their blood might experience difficulty becoming pregnant. What our data showed is that those who might have had exposure to certain PFAS, they had about a 40% reduced chance to become pregnant for 1 year, Dr. Damaskini Valvai, an author of the study in Science of the Total Environment and assistant professor of environmental medicine and public health at Icahn School of Medicine at Mount Sinai, tells Today.com. Even when we evaluated in our study lower exposure, we saw an association between people's exposure to PFAS and fertility. This is very alarming, she added. PFAS and fertility. For the study, Valvai and her team collaborated with researchers with the Singapore Preconception Study of long-term maternal and child outcomes. Their colleagues collected blood samples from 382 women, ages 18 to 40, trying to conceive. The researchers examined the levels of PFAS in their blood and their likelihood of becoming pregnant within a year. People are considered to have infertility after a year of trying to become pregnant by natural means and having no success. Study participants with PFAS in their blood had a tougher time conceiving. We were not surprised by our findings, Valvai says. There are no actual safe limits for PFAS. In populations where exposure has been substantially less, we, still, see exposure to low doses poses a risk to our health. Other studies have linked high exposure to PFAS to increased risk for some cancers, such as kidney and testicular cancer, increased cholesterol and blood pressure, and chronic liver disease, Valvai says. They're also associated with endometriosis and polycystic ovarian syndrome, which are known risk factors for infertility in women. Studies have shown that PFAS can alter reproductive hormones, and this is what inspired us to study their association with fertility outcomes in women, Valvai says. What are PFAS? PFAS, per, and polyfluoroalkyl substances are a very large class of highly persistent chemicals that have been used in consumer products and other industrial applications for more than 60 years, Valvai explains, adding that the U.S. Environmental Protection Agency has identified more than 20,000 chemicals as PFAS. The reason why we refer to them as forever chemicals is that they have many carbon-fluorine bonds that are very persistent and hard to break down, Valvai explains. Once these chemicals are used in our environment, leading to human exposure, we know they can stay there for several years. PFAS can be found almost everywhere, with Valvai noting there is global contamination. PFAS exist in drinking water and consumer products, including cosmetics, stain-resistant products, and non-stick cookware, she says. There are certain things we can do to reduce our exposure, but unfortunately we cannot completely avoid it, she says. It's hard to know where PFAS are found. If you're concerned about your fertility and PFAS exposure, Valvai recommends using certified water filter that removes PFAS, avoiding takeout and fast food because PFAS can be found in the containers, using stainless steel pans, and avoiding stain-resistant and water-resistant items. Alone, we cannot completely avoid that exposure, she says. What we actually need to be able to effectively address the bigger problems is getting stricter regulations that ban the presence of PFAS in consumer products and those in drinking water in the US and also globally. This article was originally published on Today.com. These chemicals, found almost everywhere, can reduce fertility by 40%. Study finds copyright provided by Today. Continue reading. Researchers treat consequences of brain injuries with nasally administered stem cell secreted factors. Health topics mentioned in this article: traumatic brain injury, inflammation, injury. Brain injuries cause widespread damage to brain tissue. Damaged cells begin to produce signaling molecules that trigger processes of inflammation, apoptosis, programmed cell death, and oxidative stress. After the acute phase, about a day, chronic neuroinflammation occurs. It can last a long time and cause secondary damage with negative consequences, including behavioral and cognitive dysfunction. RUDN University doctors have shown that glial cells help mitigate neuroinflammation due to brain injuries. More precisely, it is the small bubbles they produce, vesicles, which carry proteins and other compounds. Glial cells release a wide range of physiologically active substances in extracellular vesicles to support neurons. We investigated whether it is possible to use extracellular vesicles of glial cells to protect neurons in a model of traumatic brain injury, Diana Salikova, PhD, head of the Laboratory of Cellular Biotechnology of RUDN University, said. Doctors experimented on 30 adult rats. The animals were surgically subjected to an artificial brain injury, after which 15 animals were intranasally injected with extracellular vesicles of glial cells after the injury. The glial cells themselves were obtained from induced pluripotent stem cells from healthy donors. A week later, RUDN doctors checked the condition of the animal's brain tissue using PCR analysis and Western blotting. After another week, the authors conducted a series of tests to determine the behavioral characteristics of the animals. The damaged area in animals receiving vesicles decreased slightly by only 4 cubic millimeters. However, the therapy contributed to a significantly faster recovery of the animal's brain functions, improving the sensor-motor functions of the damaged hemisphere and reducing inflammation. For example, the number of CD68 plus macrophages responsible for inflammation decreased threefold after therapy. The level of the caspase enzyme, which triggers apoptosis, decreased by 2.5 times. RUDN University doctors also discovered a molecular mechanism that explains this effect. Supposedly, 203 types of small non-coding nucleotide sequences, microRNAs, became agents of neuroprotective action. Extracellular vesicles from glial cells have shown therapeutic effects in traumatic brain injury. They stimulated nervous system recovery and neuroplasticity. In addition, we found 203 types of microRNAs, which apparently can be used to treat various brain pathologies, Diana Salikova, PhD, head of the Laboratory of Cellular Biotechnology of RUDN University said. More information. Diana I. Salikova et al. Extracellular vesicles of human glial cells exert neuroprotective effects via brain miRNA modulation in a rat model of traumatic brain injury, Scientific Reports. 2023. DI 10.1038/S41598-023-47627-2. Provided by RUDN University. Unprecedented level of danger, meaning explored as atomic scientists' doomsday clock is set at 90 seconds to midnight. Add. Follow. Story by Karishma Rao 8H. The Bulletin of the Atomic Scientists have set the annual doomsday clock's hands at 90 seconds to midnight. Scientists claim that the clock's hands have shifted drastically due to the impending danger of a new nuclear arms race, climate change, and the threats of artificial intelligence. In a post on X, the Bulletin of the Atomic Scientists page uploaded a picture of the clock with the caption, "Today, the Bulletin's Science and Security Board once again sets the hashtag Doomsday Clock at 90 seconds to midnight. Humanity continues to face an unprecedented level of danger." The Doomsday Clock is a symbolic timepiece that warns humanity about edging nearer to the destruction of the world with dangerous technologies of our own making. The organization revealed on Tuesday that China, Russia, and the US were spending lump sums of money to expand or modernize their nuclear arsenals, which pose a risk of nuclear war, either by mistake or miscalculation. The bulletin also shared that the war in Ukraine, alongside Israel's nuclear arms potentially being used in their conflict with Gaza, played a role in the change in the clock. Scientists also cited the lack of action on climate change and the risks of misusing biological technologies and AI tools as reasons for the change in the doomsday clock. Who created the doomsday clock revealed as timer inches closer to midnight? According to the Bulletin's Instagram page, the Doomsday Clock was created by artist Martel Langsdorff after being asked to make a cover design in 1947, 2 years after it was founded by scientist J. Robert Oppenheimer and fellow experts upon seeing the devastating effects of the atom bomb on Hiroshima and Nagasaki. They wanted to put pressure on global leaders to ensure that nuclear weapons were never used again. The bulletin revealed that their Science and Security Board set the clock annually. According to BBC, the hands of the clock have been moved 25 times so far. It was first moved in 1947 as it was set 7 minutes until midnight. Following the end of the Cold War in 1991, the clock was set 17 minutes until midnight. The clock remained at 90 seconds until midnight, both in 2023 and this year, as per the bulletin. Speaking about the possibility of Russia and Israel using nuclear weapons and it posing as a threat to the world, Rachel Bronson, the Bulletin's president and CEO, said in an interview with Reuters. A durable end to Russia's war in Ukraine seems distant, and the use of nuclear weapons by Russia in that conflict remains a serious possibility. In the past year, Russia has sent numerous worrying nuclear signals. As a nuclear state, Israel's actions are clearly relevant to the doomsday clock discussion. Of particular worry is that the conflict might escalate more broadly in the region, creating a larger conventional war and drawing more nuclear powers or near-nuclear powers. BBC revealed that despite the presence of numerous arms control agreements, there are about 13,000 nuclear warheads globally, with the majority being in Russia and America. Speaking about the concerns of global warming, Bronson noted, "The world in 2023 entered into uncharted territory as it suffered its hottest year on record and global greenhouse gas emissions continued to rise. Both global and North Atlantic sea surface temperatures broke records, and Antarctic sea ice reached its lowest daily extent since the advent of satellite data." According to News 18, $1.7 trillion were invested into the creation of clean energy, yet the efforts in mitigating climate change are still grossly insufficient. Unprecedented level of danger, meaning explored as atomic scientists' doomsday clock is set at 90 seconds to midnight. Copyright provided by SK Pop. Continue reading. Ad. Visit SK Pop. Military intelligence. Cyber attack on Russian Scientific Research Center deals devastating damage. Ad. Follow. Story by Martin Fornasek, 4H. Volunteer Patriot, hackers of the BO team launched a cyber attack against the Russian Far Eastern Research Center of Space Hydrometeorology, Planeta, destroying its database and expensive equipment, Ukraine's military intelligence agency, HR, said on January 24. Cyber attacks are a common tool used by both sides of the Russia-Ukraine war. In one of the most devastating strikes, Russian hackers targeted Ukraine's telecommunications provider, Kyivstar, causing communication outages nationwide last December. Media reports on the cyber attack against the Russian Space Hydrometeorology Center emerged already on January 23. Ukraine's military intelligence agency now confirmed the reports, saying that the hackers allegedly destroyed 280 servers and 2 petabytes of data. Some of the lost meteorological and satellite data was actively used by the Russian Defense and Emergency Situations Ministries, the Roscosmos Space Agency, and other governmental departments, according to the Military Intelligence Agency. The center reportedly provides data to over 50 state entities. The center also lost a digital array worth at least $10 million, the agency said. The attack further destroyed the software of the center's supercomputers, each worth $350,000, effectively disabling them beyond repair. Next to the lost data and equipment, the hackers also allegedly disabled the station's air conditioning and humidification systems, as well as the regulation of emergency power supply. The attack is also said to have cut off a Russian Arctic station on the Bolshevik Island, carrying out important tasks in the military sphere. In summary, dozens of strategic companies of the Russian Federation, working in defense and playing a key role in supporting Russian occupation forces, will remain without critically important information and services for a long time, the Military Intelligence Agency said. The research center lies in the Russian far eastern city of Khabarovsk, positioned some 30 kilometers from the China-Russia border. Operated by the state-owned Rosadramat Hydrometeorology Agency, the Far Eastern Station is the largest of the research center's 3 branches. The center's main activities include receiving and processing data from Russian and foreign satellites, according to its website. The website makes no mention of the reported cyberattack. Read also, massive cyberattack targets Ukrainian online bank. Chemicals in water and household objects could reduce chances of pregnancy, live births. New study. Ad. www.nasdaq.com/board_directors/portal trusted board portal software, board portal for directors. Ad. www.nasdaq.com/board_portal/request_demo all-in-one board management, trusted board portal software. Follow. Story by Melissa Rudy, 10mo. Health topics mentioned in this article: infertility, eclampsia, reproductive health. For women who are struggling to conceive, chemicals could be the culprit. That's according to a new study published in Science of the Total Environment. The study found that women whose blood plasma contained higher amounts of PFAS, perfluoroalkyl substances, chemicals often found in drinking water and in ordinary household products, had up to a 40% lower chance of pregnancy and live births. Researchers from Mount Sinai, an integrated health system that includes the Icahn School of Medicine and 8 hospital campuses in the New York metropolitan area, focused on 1,032 Singaporean women between the ages of 18 and 45 who participated in a population-based study. COVID-19 vaccines not linked to fertility issues, experts say. Within 1 year of follow-up, the women with a combination of 7 PFAS in their blood were 30% to 40% less likely to conceive and deliver a live birth. Out of all types of PFAS, the one that had the most adverse impact on fertility was perfluorodecanoic acid, PFDA. Read on the Fox News app. PFDA is found in the environment and also in coatings on carpets, couches, and food packaging, according to the National Library of Medicine. Other chemicals that affected fertility in the study were perfluorooctanosulfonic acid, a key ingredient in stain repellents, perfluorooctanoic acid, found in Teflon and Gore-Tex, and perfluoroheptanoic acid, an environmental contaminant. The study's senior author Dr. Damaskini Valvai, assistant professor of environmental medicine and public health at Icahn Mount Sinai in New York City, believes that PFAS pose multiple risks to public health. Sperm counts and concentrations declining globally since 1970s, but fertility implications unknown. Study. In girls and women in particular, PFAS exposure has been further associated with delayed puberty onset, as well as increased risk for endometriosis, PCOS, and breast cancer, which further supports the adverse effects that PFAS have on the female reproductive system, he told Fox News Digital in an email. PFAS have also been shown to increase the risks for thyroid diseases, high cholesterol and blood pressure, diabetes, and chronic liver disease, as well as certain types of cancer, such as kidney and testicular cancer, Dr. Valvai added. Dr. Kelly Johnson Arbor, medical toxicologist and interim executive director at the National Capital Poison Center in Washington, D.C., said PFAS exposure is also associated with increased risks of gestational diabetes and preeclampsia, as well as decreased fetal growth and reduced birth weights. She was not involved in the study. Maternal death rate is on the rise in the U.S., the CDC reports. Since PFAS chemicals are known to cross the placenta, they can potentially affect the health of both the mother and the fetus, she told Fox News Digital in an email. Lead study author Dr. Valvai noted that population studies in the US and many other countries have identified adverse health effects from exposure to even very low amounts of PFAS. There is also continuous PFAS exposure through the consumption of contaminated drinking water, contaminated food, and consumer product use, which means PFAS accumulates in our body over time, potentially increasing our risk for chronic diseases in later life, he said. At the current time, we don't have enough data to tell us what a safe level of PFAS exposure is, said Dr. Johnson Arbour. There are ways to monitor the levels of PFAS in human blood, but these tests are not readily available in hospitals or doctors' offices and are typically used for research purposes only. Dr. Valvai stressed the importance of limiting exposure to PFAS, given the health risks. However, because PFAS are widely used in several consumer products and we now face a global contamination PFAS problem, we cannot fully avoid exposure without stricter regulations that ban the use of PFAS as a class, he told Fox News Digital. There are more than 10,000 PFAS released in our environment, but current regulations consider only 10 to 20 of those. That's not enough. To effectively address the PFAS problem, it is important to advocate for stronger regulations that ban the presence of PFAS as a class in consumer products and drinking water in the US and globally, Dr. Valvai continued. US to propose restrictions on forever chemicals in drinking water after previously underestimating its risks. Earlier this month, the Environmental Protection Agency, EPA, proposed new federal regulations of PFAS chemicals in drinking water, which would limit them to the lowest possible level that can be measured. The proposal calls for limiting the PFOA and PFOS chemicals to 4 parts per trillion and would also limit 4 other PFAS as a mixture. EPA's proposal to establish a national standard for PFAS in drinking water is informed by the best available science and would help provide states with the guidance they need to make decisions that best protect their communities, said Michael S. Reagan, EPA administrator, in a press release on the agency's website. This action has the potential to prevent tens of thousands of PFAS-related illnesses and marks a major step towards safeguarding all our communities from these dangerous contaminants. Based on the results of the study, it is reasonable to recommend avoidance of excessive PFAS exposure for women who are trying to become pregnant or who are experiencing infertility, said Johnson Arbour. With PFAS present in everything from cookware and carpets to drinking water and the air we breathe, it can be difficult to completely avoid exposure. However, there are some steps people can take to reduce the risk. Dr. Valvai recommends using a certified water filter that removes PFAS in the home and also using stainless pans instead of nonstick cookware. It's best to avoid stain-resistant and water-resistant products because almost all of these products contain multiple PFAS, he added. Scientists reveal simple new process that may help eliminate toxic chemicals from everyday items. Study co-author Dr. Nathan Cohen, a postdoctoral research fellow with the Department of Environmental Medicine and Public Health at the Icahn School of Medicine at Mount Sinai in New York City, told Fox News Digital that diet is a very common source of exposure to PFAS. Women who are planning pregnancy should avoid foods that have been shown to be associated with increased PFAS exposure in previous studies, he said. These primarily include takeout and fast foods that are packaged in materials that may contain PFAS, such as fast food, popcorn, and pizza. Fried foods, such as fried fish, may also pose a risk and should also be avoided. The researchers noted that one major limitation of the study was that they did not have data available on PFAS concentrations in the male partners in terms of fertility impact. Click here to sign up for our health newsletter. We also did not have data available on reproductive hormones to assess hormone disruption as a potential mechanism, Dr. Cohen said. Additionally, we focused our PFAS assessment on those detected at high concentrations in other populations, so we were not able to assess the effects of emerging PFAS. The study was funded by the National Research Foundation via a grant from the Singapore Ministry of Health's National Medical Research Council and the Agency for Science, Technology and Research and the U.S. National Institute of Environmental Health Sciences, according to the press release from Mount Sinai. PFAS, perfluoroalkyl substances, are chemicals that are often found in drinking water and ordinary household products. iStock Copyright iStock. Women with a combination of 7 PFAS in their blood were 30% to 40% less likely to conceive and deliver a live birth, according to new research. iStock Copyright iStock. To avoid PFAS exposure, the study author recommends using stainless pans instead of nonstick cookware. iStock Copyright iStock. Of all types of PFAS, the one that had the most adverse impact on fertility was perfluorodecanoic acid, PFDA, which is found in the environment and also in coatings on carpets, couches, and food packaging. iStock Copyright iStock. Related video, Gravitas, is your plastic water bottle killing you? Wyan. View on watch. Continue reading. New technology allows researchers to track brain cells off switches. Ad. Fisher Investments 13 mistakes investors with $1 million make and ways to avoid them. Ad. www.nasdaq.com/board_directors/portal board management software used by 4.5K plus global companies. Follow. Story by Science X Staff 23H. Health topics mentioned in this article: Alzheimer's disease, weight loss, post-traumatic stress disorder. For decades, scientists have studied the intricate activity patterns in human and animal brains by observing when different groups of brain cells turn on. Equally important to understanding the brain and related diseases, however, is knowing how long those neurons stay active and when they turn off again. Now, scientists at Scripps Research have developed a new technology that lets them track when, after a burst of activity, brain cells shut off, a process known as inhibition. The technique, published in Neuron, provides a new way to study not only the normal functioning of the brain, but how the brain's off switches may go awry in normal behaviors as well as in diseases and disorders, including depression, post-traumatic stress disorder, and Alzheimer's disease. The paper is titled Phosphorylation of Pyruvate Dehydrogenase Inversely Associates with Neuronal Activity. It's generally agreed that the inhibition of neurons is really the major way the brain is regulating activity, says senior author Li Yi, PhD, professor and Abide Vividian chair at Scripps Research. Scientists have been looking for a way to look at inhibition in a more trackable way, and until now, few had found it. For pioneering the new approach, Yi teamed up with John Yates, a professor of molecular medicine at Scripps Research. They wanted to study how brain cells changed when they were actively firing, emitting an electrical charge to pass messages to their neighbors, compared to when they were done firing. The scientists used optogenetics, in which cells' activity can be controlled using light, to repeatedly activate and inhibit the cells. Then, they measured levels and characteristics of different proteins and their modifications. They identified that one protein, pyruvate dehydrogenase (PDH), was very rapidly changed immediately after brain cells were inhibited. When neurons are firing, you need a lot of energy, and this PDH protein is involved in producing that energy, explains Yi. But the brain really wants to conserve energy, so when a cell is done firing, we found that the brain rapidly shuts off the PDH protein. This happened much faster than anything else we saw in gene expression. To shut off PDH, the researchers found cells add molecular tags called phosphates to the protein. Yi and his colleagues found antibodies that only recognize this inactive, phosphorylated form of PDH, PEPDH. To test whether levels of phosphorylated PDH or PEPDH could be used as a proxy for brain cell inhibition, Ye's team used these antibodies to measure PPDH in mice that had been given anesthesia. Nearly the entire brain lit up with high levels of PPDH, correctly showing how most of the brain is inactive during anesthesia. The group also studied levels of PPDH when animals were exposed to bright light that was then shut off. Brain cells in the visual cortex, responsible for vision, had low levels of PPDH when being exposed to light, because the active form of PDH would be required to give these cells signaling energy. But high levels of phosphorylated protein immediately increased after the light was off. Ye's group also used the new technique to study a less understood process, how the brain turns off the feeling of hunger after a meal. They showed how brain cells related to appetite shut off when a mouse starts to eat. These findings could have implications for better understanding appetite, obesity, and some weight loss drugs. More broadly, the PEPDH antibodies could be used to compare levels of brain cell inhibition in healthy people and those with a variety of brain and metabolic diseases. There are a lot of questions that this technology can help us answer, says Yi. If the brain can't turn cells off, or if they're turned off faster or slower than usual, what happens? How does the inhibition of neurons play a role in different diseases? Yi and his colleagues are continuing to fine-tune the use of PEPDH, but they say that other researchers are already using the technology. The antibodies used to measure PPDH are commercially available. More information. Phosphorylation of pyruvate dehydrogenase inversely associates with neuronal activity. Neuron, 2024. DI 10.1016/J.neuron.2023.12.015. www.cell.com/neuron/S0896-6273-23-00974-1. Provided by the Scripps Research Institute. This story was originally published on Medical Express. Scientists warn, both plastic and its alternatives are toxic and pose health risks. Ad. Joiny this headlight glasses that's making nighttime driving as daytime driving. Ad. www.nasdaq.com/board_portal/request_demo trusted board portal software, board portal for directors. Follow. Story by Migo 1MO. Health topics mentioned in this article: water, caffeine, coffee. Reports of plastic pollution affecting every corner of the globe and all living beings have provoked a transition towards alternative materials. However, scientists from Gothenburg University suggest that using paper substitutes may not be as safe and eco-friendly as initially thought. The impact of disposable cups on butterfly larvae. Researchers from Gothenburg University studied the effect of disposable cups made from various materials on mosquito larvae. We exposed both paper and plastic cups to wet sediment and water for several weeks and observed how the leached chemicals affected the larvae. All cups had a detrimental effect on the growth of mosquito larvae, explains Bethany Carney Amroth, a professor of environmental science at the Faculty of Biology and Environmental Science at Gothenburg University. Paper cups are lined with a thin layer of plastic film. Ordinary paper is not fat or water-resistant, therefore paper used for food packaging needs to be coated with a protective layer. Currently, this layer often consists of polylactide, PLA, a type of bioplastic. Bioplastics, including PLA, are derived from natural and renewable resources like corn, cassava, or sugarcane. They are considered to be biodegradable, meaning they decompose faster than traditional plastics under the right conditions. However, these bioplastics have proven to be toxic. Bioplastics don't decompose effectively once they enter the environment, such as water. there's a risk that the plastic remains in nature, breaking down into microplastics, which could potentially be ingested by animals and humans. Bioplastics contain just as many chemical compounds as conventional plastics, explains Bethany Carney Amroth. Potential health risks associated with food packaging. It appears that paper food packaging still poses a risk of exposure to harmful chemicals that can get into our food. The study's findings were published in the Environmental Pollution Journal. Besides the results themselves, the authors also discuss ways to mitigate the risk of food contamination by plastic. When disposable products hit the market after World War II, there were major campaigns to teach people to dispose of products, which was abnormal behavior. Now we need to revert, moving away from this disposable lifestyle. I suggest people bring their own cup when buying takeaway coffee. Better yet, take a few minutes to sit down and enjoy your coffee from a porcelain cup, suggests Bethany Carney Amroth in her article. Binding agreements to minimize plastic use are under discussion. The UN is currently working on an agreement to curb the spread of plastic in society and the environment. The Coalition of Scientists for an Effective Plastic Treaty, CEPT, is contributing scientific evidence to these negotiations. The coalition advocates for a quick transition away from unnecessary and problematic plastics, while being wary of replacing one harmful product with another. We at CEPT are pushing for transparency requirements in the plastics industry, demanding clear reporting of the chemicals that all products contain, similar to those in the pharmaceutical industry. However, the primary aim of our work is to reduce plastic production, says Bethany Carney Amroth. Read also. Caffeine can kill you. Scientists warn about the overdosing. Health alert: the unseen risks of sweetened Turkish coffee.